Perhaps I should introduce myself. My name is Nathalie Isabelle, and I work for the Canadian Forest Service. I've been working there for 25 years, so here we are. So now let's continue on to speak about or to talk about forest health. So with our next speaker is Jennifer Koch. And Jennifer is a research biologist with the USDA Forest Service Northern Research Station in Delaware, Ohio. She has a MS master degree and a PhD degree in molecular genetics from the Ohio State University. Her career in the Forest Service has focused on the genetics of host resistance to invasive insects and diseases, developing methods to identify, propagate, and breed resistant trees for restoration of urban and natural forests. She collaborates with national and international partners to accelerate breeding by identifying the underlying mechanism responsible for resistance. I visited Jennifer a couple of years ago, just before the COVID, and I was amazed by the progress that were made on ash. So thank you, Jennifer, to talk about this morning about how the breeding resistance to emerald ash borer can breeding prevent extinction. So thank you very much. I know how stressful it is working for the government and to get the permission at the very last minute. So thank you again.
Thank you, Natalie. It is an honor to be here for this first meeting of the North American Forest Genetics Society. I appreciate the organizers for organizing this. I think it's going to be a very important group of people. As we all know that we have our forests are facing a lot of threats, and novel insects, climate change, and these things are not sticking to regional borders. And speaking of a threat that's not sticking to regional borders, how many of you have heard of emerald ash borer? Can I see some hands? Okay, so good. The majority of you have. So I have not included a lot of background slides, but if you do have any other questions, please catch me when you can, and I'm happy to talk about it more. So to summarize, emerald ash borer is an invasive beetle that came to North America uninvited from Asia, and it was first noticed in the Detroit area as well as Windsor, Canada in the year 2002. Since then, we've learned that EAB can cause up to 99 to 100% mortality within 4 to 7 years of first seeing any symptoms. So this near synchronous loss has cascading effects on our ecosystems, degrading our clean water, degrading the aesthetics of recreational activities, altering forest composition, and even impacting human health. There's studies that have shown the loss of urban trees. Green ash was very commonly planted as a street tree, is correlated with the increase of mortality due to respiratory and heart disease in humans. So at this point, the IUCN has declared 5 of our Eastern North American ash species, green, white, black, blue, and pumpkin, as critically endangered. There we go. But fortunately, we do see some variation in the phenotypes in emerald ash borer as it moves through our genetically diverse populations of ash. Unfortunately, the most common one we see is the one on the far left, which is dead, because it's feeding on the external bark tissues through the phloem into the xylem until it's girdling the tree. However, in rare occasions, we see what's illustrated in the middle photo, where you can see evidence of damage from EAB feeding, but it has healed, and the canopy in these cases remain healthy. In extremely rare situations, we've only seen 2 of them so far, the bowl remains completely healthy, and we cannot even tell that EAB has done any damage. So we first discovered these trees, this was thanks to a lot of monitoring work by our ecology colleagues who surveyed hundreds of plots throughout Ohio and Michigan. When these surviving trees were found, we didn't want to be overzealous and refer to them as resistant until we had more data to support that. So we came up with the term lingering. So at this point, based on the monitoring work, the criteria that we use to select lingering ash trees is we're looking in areas that have been long infested by emerald ash borer. We want trees that are big enough to have been infested during peak EAB, because we know EAB has some very specific size preferences. We want to see a healthy canopy, and we want trees that have been alive for at least 2 years after the mortality rate has leveled off. So this is about 2 years after 95 to 99% mortality, which also coincides usually quite closely with about 4 years after 50% mortality. And the photos here show you that we're not talking about escapes, because these trees are found right next to other trees that have been killed by EAB. What am I doing wrong with this? There we go. So this slide is an overview of our breeding program. It's very complex, and I don't expect you to be able to read it or follow it. I just wanted to give you an idea of all that we have going on pretty much simultaneously. And in the upper left-hand corner, that's the part that I already talked about, ID and selection of trees in the field. But at the same time that we're trying to simultaneously identify these trees, select them, propagate them so that we can preserve them, because they can go on to die and we don't want to lose this valuable genetic resource. We are also trying to breed. We're doing crosses when possible. Once we graft the trees, we can get them to flower, so we can get small families, because these aren't big trees yet. We're also doing bioassays, and at the same time optimizing these bioassays. So we're sort of doing it all at the same time. We are validating bioassays by putting in field studies and then developing even new propagation methods. Right now, we've developed a rooted cutting method, which will be very useful for establishing seed orchards on their own roots, and as well as cultivar development. So the bioassay that we're focused on, that the majority of the rest of this talk will be on, is done in conjunction with our collaborators that are entomologists with the Forest Service, Therese Poland and Toby Patrice. They actually rear adult beetles, sort them into mating pairs, put them into containers, allow them to mate right onto coffee filters. They have like a screen that they put a coffee filter under, and that screen mimics like the bark cracks. So the female likes to put her ovipositor and she lays eggs, and we get these coffee filters full of eggs. We actually take measurements at this point of the trees, the diameter on the section that we're going to put eggs on, so we can calculate the dose of the eggs we put on bark area per meter square, so that we're changing the distances so that trees of different sizes are getting an equal dose, because we also have learned that this is a dose dependent response. The dose we're using is one that is high enough to mimic what you would see during the highest levels of EAP and the infestation. So after 2 to 3 weeks that we've applied the eggs, we go back and confirm whether or not they've hatched. We actually, if we can see the entry hole, we'll put a little mark around that. 8 weeks later, that is when, in a fully susceptible tree, we would expect the majority of the larvae to have reached the later of the instars, L4, which is right before prepupation and pupation. And then we get really excited when we see the one on the bottom right side, which is one that's been killed by a host defense response. When possible, we remove the living larvae, and if we haven't cut them, we collect their weights. And just to summarize, at this point, we have found significant differences in larval weights and larval development, the proportion, and as well as the proportion of larvae killed by host defense responses. We focus on that, the proportion of larvae killed, because it is the later instar larvae that cause the most damage. So that seems to be the most significant of the mechanisms that we can be looking at. And just as an aside, we also know that there's adult feeding preferences and most likely oviposition preferences that have a role in these lingering ash trees. This slide is showing the first 40 selected lingering green ash. So on the x-axis, each orange line is representative of the mean of all of the replicates of that genotype that have been tested. So there's at least 3, and in some there's a lot more. And moving from left to right, you can see that there's some trees that had 0. So even though they met our criteria, they still did not kill any larvae. So that's where those other mechanisms of preferences can come into play. And then as you're moving from left to right, you're getting to about 10%, 20%, but we're getting into that far right side, and you're hitting about 25%, all the way up to almost 50%. You can imagine that if at like 25%, 1 out of every 4 larvae or 1 out of every 2 larvae is killed, that's enough. That diminishes the amount of damage to that specific tree. That would explain why it's living longer than the rest in their population. So we know these are trees that are living longer, but they're still going to die. So that's why it's very critical that we capture them while we can. And for reference, I put on here a few black ash selections, and I'll talk about that later, but also Manchurian ash, a cultivar. So Manchurian ash is a species that originates from Asia, where EAB came from. So they've co-evolved, and it's highly resistant. So we're about halfway to the level of resistance that's shown there. At this point, we used 68 selections of green and white ash. We also had a family. We did a small cross and replicated each of those seedlings and did the EAB bioassay. And we used clonal repeatability to estimate broad sense repeatability on 365 total grafted stems. So you can see in white ash, we only had 13 genotypes, but we came up with a repeatability estimate of 0.45. Green ash was 0.15, but we had a lot more genotypes. And within family, we were at 0.87. So 0.15 may be low, but it is still reasonable compared to similar estimates of heritabilities in other tree improvement programs. And it's certainly high enough to support further investigation. We're using these bluffs from these models are being used to rank parents for priority crossing in the future, well, ongoing and in the future. And we also want to confirm the phenotypes by getting them back in the field, and this will help us assess environmental variation. So we've done this. We have 2 sites where we have replicated 40 lingering green ash and 8 white ash, as well as known susceptible controls. As I mentioned, when we graft these trees, we can get some of them to flower, so we can produce small amounts of seed. Some of our trees have now grown quite large, and we can get bigger batches. So we have now a mapping population that we're hoping is going to end up being about 400 trees. But we've started to do bioassays on these, and this is a summary of those. Oh, this is the one. I think about 20, a little over 24 families are included in this slide. Just to point out, on the x-axis is each of the individual families that are named. The y-axis is, again, the average proportion of tree-killed larvae. The green line, the green dotted line is indicating the level of resistance we found in the best selection that came straight out of the natural forest. The red line at the bottom is indicating the best performing member of an unselected family. And it's that is an unselected seed lot. And I also wanted to mention that not all of these are lingering by lingering. Some of them, including those 3 that are circled by the red box, have at least one parent that is intermediate or susceptible. The box is showing the interquartile range. The line is at the medium. The target symbol, if you can see it, is the mean, and the individual tree dots are represented, individual values for tree are represented by dots. So you can see that even in cases where we have a susceptible or intermediate parent, and even in some of the worst, we have we have variation in our families, obviously. But the really exciting news is that if you look at that green line, which is the best performing tree from a natural forest, we have plenty of progeny that are doing much better than any of the parents. So the parents are somewhere between 0.2 and that top green line. And some of the parents are up to 100% tree-killed larvae. So this is very exciting, but it has to be validated back in the field. So we're in the process of doing that. And in the meantime, we have used 27 families to date that include 12 female parents, 13 male parents, 773 total progeny that have been tested from 2018 to 2020 to calculate narrow sense heritability. And so this is a ratio of the additive variance over the total phenotypic variance. And coming out at a heritability of 0.63. And it's interesting, we did detect non-additive family effects, but at least they are smaller than smaller than the additive effects. So again, this is promising that an indication that breeding and selection is a process that can work. This is a picture just illustrating that getting the seedling families back into the field for further assessment of the phenotypes. So far, we've gotten 16 families back. That represents 14 parents and 430 progeny. We plan on culling poor performers and managing this as a seed orchard. We realize we need more genetic diversity, but at least in 5 to 7 years, we'll be able to test some seed out of this planting. Not only do we need to increase genetic diversity in our own plantings, but we need to increase it across the entire range. We need to replicate this whole process across the range if we're going to truly save ash. The seedling bioassay, what you see planted in this picture, so it's a destructive assay that's done on a single rep because we don't have grafted reps. But what we do is we leave the stump and we allow the stump to sprout, and then we train the sprout to a single leader, and then we can plant them. So these are the exact same trees that the bioassay was done on. In addition, the other sprouts are what we use to do rooted cuttings with. So we can get a lot of replication off of a single seedling once it's been cut. So finally, I wanted to address really quickly additional threatened species and range-wide restoration. To summarize, at this point, we have 65 lingering green ash selections, and they are from a limited area: northeastern Ohio, southwestern Michigan, and part of Indiana. We've bioassayed 31. We have 49 lingering families, but we've only bioassayed 25 of those. That's a huge bottleneck for us. Our capacity is about 800 trees per summer that we can test. So again, we need to increase this genetic diversity, and we need many regional, locally adapted populations. We have additional species that are threatened that I mentioned, and just to list what we've done so far, we have 29 lingering ash selections. We do have white ash families. We only have 3 black ash, 4 blue ash, and we've had reports of pumpkin ash, but we've never successfully been able to propagate them. So the other thing we've now switched to, especially with black ash, is learning, it's so closely related to Manchurian ash, is trying to figure out if the variation in the response to emerald ash borer may differ. We can't assume that everything is going to be the same as green ash just because it grows in Eastern North America. So we screened a small number of seed lots, and these are the results. So these are unselected seed lots, and it's the same proportion tree killed. The different families are listed on the x-axis, and the green line that's going along the top is again giving you the point where the best selected green lingering ash was. The blue is the best unselected. Highlighted in blue is a single family, and you can tell that there is variation across the families, and there's also variation between the families. So this means that there's the potential to screen open pollinated families ahead of EAB, because we have many areas where EAB has not yet completely devastated our black ash populations, which is going to be critical to climate change because there's about 300 million acres that risk being converted once that does happen. We did the same thing in blue ash. Oh, the bad news though with black ash is that high tree killed larvae doesn't always equal better survival. And this is just illustrating that. If you look at the black ash, you can see that around the gallery, there's this darkening color, which is indicative of a defense response, but it has expanded to the point that it's almost girdling that little seedling. So we're hoping to apply some genomics to help us better select for that. But we have done one-year bioassays and been able to select for black ash that have higher tree kill and also survive. We did a similar experiment with blue ash and we got similar results. Again, one of them is killed 100%, but there's variation between and within families. So screening OP families, rather than waiting for black and blue ash to all die, seems to be a promising method to go. So really, just to summarize, our data indicates genetic gain is possible from breeding and selection. We're getting progeny with resistance levels similar to the EAB resistant Manchurian species. We're in the process of confirming this in field trials. Proactive screening with OPC may be possible. Also looking at Oregon ash, I think Richard might mention that in the next talk, but we also need to accelerate breeding and expand our range-wide efforts. So we have been expanding through partnerships. We have something called the Great Lakes Basin Forest Health Collaborative. We're working with multiple arboreta who are helping us, Ecological Research Institute, if you want to look at monitoringash.org for the Tree Snap app. And finally, we recently were awarded significant funding from the Manton Foundation in partnership with the Nature Conservancy for a project called Trees in Peril, which will support further work to develop methods to accelerate breeding. That's traditional methods as well as application of genomics. So with that, I can't possibly acknowledge everybody who's helped with this project, but I do at least want to throw up the slide there and acknowledge all the funding sources that we've been able to get. And it looks like you've been standing there for a while, so I assume I may not have time for questions, so I apologize.
We may have time for one question. Yes, Justin?
Hey, Jennifer, nice talk. I've been seeing your work on EAP for almost a decade. So I guess my question is, when I worked with back in the 2010s, green ash resistant mechanisms seem to focus around herbicide knowledge, one of the major things that differ. And really quantitative defense, the more the compound, the more the impact on insect survival. I guess my question is are there other traits that are contributing to the phenotype that you're seeing? And if there aren't, is there any concern that having a single compound breeding into the program, you're going to put a lot of selection pressure on the insect? And if you have this resistance and you get it out, and then maybe in 2 to 3 generations, the insect evolves the amino acid change in the cytochrome P450.
Yep. Yeah, I focused on the breeding for this talk, but we have done a lot of other work with collaborators at Notre Dame University. Jean Romero-Sieberson and her grad student, Robert Stanley, worked on 3 of our largest families with our widest distributions, and they did a non-targeted metabolomic study. They did specifically look at herbascicide. They identified sets of compounds that were predictive of resistance. I think I mentioned this is a paper that's in review. But even in cases where the families had a shared parent, they weren't always the same set. And it was the quantity of these compounds that were the difference. So anyway, the bottom line from that big metabolomic study is that it isn't a single compound. It is going to be a complex trait. And that indicates that it is going to be a very complex trait. So we're looking at sort of traditional, complex, quantitative tree improvement.
I'm going to stop you just okay, we'll wait because we will have time still on during the panel discussion and
and I'm around. So.
Yeah. Thank you. So we thought it was really important to have people from the U.S. Forest Service here today. So actually, we will have Richard Sniesko. Richard is Dr. Sniesko is a center geneticist at the USDA Forest Service, the RENA Genetic Resource Center in Oregon. His work focuses on development of population of trees with genetic resistance to non-native forestry disease and pests, as well as genetic variation and conservation. I thought for my career that you were a forest pathologist because we've never met, perhaps we met once in Chicago and that's it. I never thought that you were a forest geneticist. So now he has worked extensively with white pine blister rust, Port Orford cedar root disease, and Koch wilt, among others. Some additional recent projects involve Pacific madrone, Oregon ash, and True Fires. He is the coordinator of the UFO breeding genetic resources of white pine needles pines. I've never been allowed to go there. And a deputy coordinator of resistance to insect and pathogens. So welcome, Richard. And your talk is about forest health, successes in breeding for resistance to non-native pathogen, and using sentinel trials to provide future information. And thank you, Richard, for having accepted to come here today because I know that you had another meeting in parallel. So thank you again for accepting to come here. Oh, I will let yeah.
Hello, and I want to thank the organizers for the invite to present here. I think this meeting is a great idea. I was at a Society of American Foresters meeting back in 2015 or whatever it was with David and a few other people, and Dave and others were talking about this concept. So I'm great to see it follow through. I think it's much needed now with climate change, with invasive species coming forth and killing a lot of our trees. You've already seen from the East Coast some programs. What I'm going to talk about is mostly the West Coast and Hawaii and programs that I'm involved in. Now, I come at it from a different point of view. I'm not in the research branch of the U.S. Forest Service. I'm kind of in the National Forest Branch implied thing. So my duty is to make it happen and find resistance. And I work with some folks in Hawaii with Koch, and I talked to the one gentleman there, and I told him, well, look, we can solve your problem, but I won't be able to tell you why it works. And that's where a lot of partnerships and with a lot of you can help out understanding why it works. But fundamentally, I'm involved in tree breeding and solving a problem. Okay, so I'm going to talk about today's successes in breeding. Okay, so there are successes that need to be known to non-native pathogens. And then also a second project that I started last year with a couple of people on using sentinel trials to provide future information to be ahead of the curve. What are the next threats in North America of insects or pathogens? So as we all know, there are pathogens and pests around the world that affect our trees. They can kill 99% of some of our tree species. You can try to stop them. You can try to keep them out of the country. A lot of that fails once they get in there. It comes down to genetics. And it's a very powerful tool to be harnessed, but oftentimes it gets looked at too late. People wait too long. You know, we should put it up front. And so what are the threats? You know, there's an increased public awareness that there's a threat to our forest trees. It can lead to major changes in forest ecosystems. It can lead to serious economic losses, and then climate change will complicate all of this. I know there's a discussion session after this. I'm going to go through things very quickly, show you a lot of what's been done without going into detail, and we can talk more later. So again, there's, you know, I'm a big proponent on keeping new pathogens and pests out of the country. That should be number one, no doubt about it. But in cases where that fails, ultimately it will come down to genetics, and that's where a lot of you can be involved and be helping with these programs. A key is whenever you're working with a program, Jennifer and I published this little diagram in a paper in 2017 and whatever like that too. This is key to remember. There's a research phase to things, but there's also a tree improvement and tree breeding phase. You have to have management commitment over time, and you have to have restoration and reforestation. And I like to tell anybody that if you're in one of those and all four of them aren't there, you're not going to have success. If you're in research and you discover a couple of genes for resistance, but nobody's using it, well, hey, so what? If I'm in tree improvement and I develop the best resistant material and silviculturists aren't planting it, so what? If you don't have management support, the program's not going to be successful. Right now with Porterford cedar, we have an opportunity. You'll see a little bit about the program in a minute. I think it will potentially be the first species in the world potentially that's been on a kind of a threatened list that will be functionally restored. And we have tribes, federal agencies, non-industrial landowners, a lot of people interested. We have the product. I told them now, I had a meeting with them in March out in the field and said, now it's up to you guys. The other three groups have done their part. Now it's up to the land managers. This is a photo of Drano. We do things on the large scale, okay? And, you know, we have a ton of resistant material. So if you need well-defined resistant material for certain systems, we test it, we retest it, we put it in the field, and, you know, we play devil's advocate and say, hey, are we sure this works? And we have some very high performing stuff. We probably have the largest fog chamber in the world for this type of work. So again, we do things on a very, very large scale. Resistance is a numbers game, especially to non-native invasives. This is a list of some of the species in the West that have resistance programs at different stages. A lot of the ones at the top are 5-needle pines and white pine blisteras, you know, classic. And depending which of the 8 Western species you're working at, we're at different stages there. There's different types of resistance. I'll show that in a minute. Porterford cedar is a real success example, I think. Koa over in Hawaii, I was asked to go over there in 2003, and now there's a high-end guitar maker bought land over there, putting in plantings, and you know they're deeming it a success. You know, so we'll see. And then ohia over there, the really keystone species over there is really threatened by actually 3 diseases. Some of them more virulent than the others, and so we just started a program with that. So, you know, phase 1 considerations, is there genetic resistance? You hear people say at times, well, no, I don't think there's any genetic resistance. And I was asked at a panel once for National Academy of Science when I was giving a webinar. So, hey, do we need to do genetic engineering for any particular species? Have you found there's any species there's no resistance for? And I said, I can't think of a species that there hasn't been shown definitively there's no resistance for. I can see people have made that statement, including about one of the species I will talk about, Porterford cedar. For 15 or 20 years, a pathologist worked with Porterford cedar looking for resistance. It's in the literature that there is no resistance to Porterford cedar. Then they were about to give up. They had a couple clones left. They decided to retest them one last time. And in 1989, they published a paper saying, by golly, there is some resistance. They kind of mischaracterized the resistance. But from that point, the U.S. Forest Service then took off and started an operational program to develop resistance, and I'll show you some of those results. So you have to be careful when you're looking at resistance to look at it properly and not overstate it, not understate it. So what's the frequency and geographic distribution of resistance? What type of resistance? Major gene, quantitative? What's the level of resistance? Is it durable? Will it last forever? Is it stable? And I think you also got to know that trees are different than crops. You know, you can take shortcuts of crops to develop one cultivar. I found a Porterford cedar that had 100% resistance, and one of the pathologists said to me, hey, your job's done. You just cross out one parent with every other tree. And I said, well, not quite that simple. And so, you know, maybe with crops, and if we were interested in cultivars, we could do something like that. Kind of a phase 2, you want to know, is breeding necessary? For some species, yes, definitely. For some species, no. I think we have enough resistance in the first generation with enough phenotypic selection that we lucked out. Do we need seed orchards? Can we go back to the parent tree? How do we organize a successful applied program? Because when you look around the world, there aren't that many of them. I would venture that for non-native invasives, probably 3 of the top 5 programs in the world that are our facility. And there's a reason for that Venn diagram that I showed you. You know, how to maximize restoration, that's the real key, getting it back out on the ground once you have a resistance. And then the fundamental program, all the programs to date, for the most part, have been developed without using genomics or mass phenotyping. And there's various reasons for that. But I'm real excited about the potential that there is there. And so we have to look in a way. There's so many species affected by these things, we have to find shortcuts. So I think genomics and some of the other tools will allow us to do that. Here's white pine blisteras. Most people know the story, alternate host. It can kill 99% of your white pine species in some areas. There's major gene resistance and there's quantitative or partial resistance. Okay, that's kind of the story there. And it somewhat varies by species, both which ones, not all species appear to have major gene resistance. And some species appear to have quite a bit of quantitative resistance. Others have almost nothing. We're looking at foxtail pine now, a range-wide study of 150 families. And I'm hoping that there'll be 10 seedlings left out of, you know, the 6,000 or so that we have out there. It's very, very susceptible. This is the range of the white pine or 5-needle pines. On the left-hand column, the number, again, it's a numbers game. Pinus lambertiana, Pinus monticola, we've screened seedling families of over 5,000 parent trees, you know, and probably the best parent tree other than major gene resistance for quantitative resistance we found for sugar pine might give us 10% survival in our test. So you're starting at a low level. White bark pine, we've screened a lot less, but we've already found a number of them that will give us 50% survival in our tests and stuff like that too. So each species can be different. You have to be careful generalizing. This is a field test of Western white pine, and 13 families were put out. And I apologize if anybody's colorblind because I use green and red in here, and these are some old graphs and they're redone, and I just didn't have the new ones ready. But for those of you who can see it, the red line is a susceptible, both for percent stem symptoms or cankering on the left and percent mortality on the right. And you can say, hey, great, Rich, you've got a good test. Your susceptible does really bad, like it's supposed to. But you have a couple lines that are above the susceptible. What are those? The ones that are above the susceptibles are actually our new susceptibles. They're the trees with major gene resistance, okay? Because a virulent strain of the pathogen has arisen, and in Western Oregon, if you plant Western white pine with major gene resistance, it's going to look worse than our susceptible. And there's a couple of reasons for that, and you know we can talk about that in the discussion period and stuff like that too. But it's technically worse than our susceptible. All the families out there that are doing well right now, and some of these trees are pretty big. It was planted in 1999. You know, some of them have cankers and are living. Some of them don't have cankers within the same family. You know, nature of quantitative resistance. This is white bark pine. These are Chintry row plots in these boxes. You know, our statistician passed away a couple of years ago of cancer, but I used to kid Doug. I don't need to tell me that hey, the green row is different than the red row. You know, I don't even need statistics on that. And so we lucked out with white bark pine. Before we started the program, some of the literature said white bark pine was the most susceptible of all the species. Well, it kind of depends what you mean by susceptible. The susceptible individuals within white bark pine are very susceptible, but there's more resistance in white bark pine than we ever found in Western white pine or sugar pine. Here's kind of a map with pie clusters of where do populations vary in resistance. And long story short here, the ones with a lot of green and blue in the clusters have a lot of resistance. The ones like on the Olympic Peninsula there that have red and orange essentially have very little or no quantitative resistance. And so far we haven't found major gene resistance within white bark pine, but we do have one family that has 90% survival in our test, which is off the charts, even to the second place one. And so maybe with some genetic genomics tools and various other things, we can figure out both. So is that major gene resistance or what's going on? Success stories. This is if you go to Crater Lake National Park, they have 6 restoration plantings out. This is one of them. This is at Rim Village. 400,000 people a year see this planting. Everybody at the park by Rim Village and stores, they walk by this planting. Some of the trees are now 6 feet high. So time will tell what happens, but we have some resistant families in here and some susceptible ones. There is some blister rust in there, but a lot of field trials, it takes a while before them to get high enough incidence. You know, I've recently talked to some of our people in Eastern Washington, and I said, I want your worst site for blister rust. I want to plant some stuff out there. I want results in 5 years, not 15 or 20 years. But this would be a cool trial because the public will see it. There's a chance for conservation, education, et cetera, et cetera. Porterford cedar, switching. Porterford cedar gets a root disease. There were lawsuits against the Forest Service and BLM. I told you the resistance story when they first declared there was no resistance. There was found to be some resistance. We were asked to start a resistance program. We started it. Within 5 years of starting the resistance program, we had our first resistant seed out for the first seed zone or breeding zone and stuff like that too. We're since developing higher levels of resistance over different breeding zones and various other things. So not all trees have to take forever. Now, if we have some white bark pine has an advantage. I'll show you in a minute of the materials and what you can do. White bark pine, you can do rooted cuttings, you can do self-pollination, you can get it to flower at age 1 or 2. And at Dorena Genetic Resource Center, we probably have 1,000 or 1,200 parent trees from sea level to 5,500 feet, probably the most complete collection of a species maybe in the world for a conifer. So now, again, I'm not a research unit, so there's all sorts of stuff that can be done with that material. So for the right researcher out there that wants to cooperate or look at something and wants to look at a model species, Porterford cedar could be the one for you. So our job is to develop orchard population Porterford cedar that have durable resistance and get it back out there. And that's what we're starting to do for some of the breeding zones now. Porterford cedar used to be a big, massive tree. 2 years ago, I was called by one of the tribal elders in Koch tribe. And he says, Richard, I've heard about this resistance. I want the big trees back. Okay. They also have some land that they grow for timber, but he was more interested in the land that they use for restoration and getting the big trees back. And so I said, oh, okay, hey, What I'll do, I can tell you where to get seed because we can't, for general reforestation. But I said, if you're willing to set up a joint genetic trial restoration, we'll grow the seedlings, we'll label them, you guys plant them, will be a joint project. So I think back in March, 1,000 or so seedlings went in a trial on this tribal land. So we'll see how it goes. And I think theirs will be one of the better sites because the site prep on their land is better than it is on Forest Service land. The amount of resistance and stuff like this is a really early graph and stuff like that too. But for Porterford cedar, you can get, 50, this is in the greenhouse test, 50% or so survival versus maybe less than 10% survival for Woods Run. So, hey, it's really ready for prime time. We're not talking about a cultivar where you're looking for 100% survival. That'd be great, but we're also maintaining genetic diversity and genetic adaptability. People are planting it out, both industry, tribal, volunteers to protect the water with shade and whatever like that too. So it's getting out on the landscape. On the road to success, in the year 2000, this was rated by an international group as vulnerable, Porterford cedar. And then they downgraded it to threatened as of 2013. And they said if the federal agencies and the landowners keep up what they're doing, we think that within 10 years, it will be a species of least concern. So my message when I was out in the field and gave a webinar in February to these land managers, I said, it's in your court now. I said, you guys could be part of the process that makes this perhaps the first species in the world to be taken off of a list like this from a non-native. So tree breeding is the key, but is biotech useful without tree breeding improvement? No, you really need to be coupled with the people that are doing the tree improvement, the breeding. And then Dave already talked about the decline in genetics and tree improvement across the U.S. It's something that we've got to reverse to a certain extent. And I know there's been a number of hirings at universities and within Forest Service recently. So I'm somewhat optimistic that we'll go at least a little bit upward in that regard. What we do need is it still takes too long, even with the screening that we do, and it's expensive. We can do it. I think almost every species can do it, but we need better tools. And that's where genomics and other genetic studies and mass phenotyping and everything can come in there. My dream is to have a tricorder where I can walk out to a tree and tell you whether it's a good candidate for resistance or not. So how we get there is, much smarter people than me. So wish list, we want to find what is durable resistance. When that was first coined by Johnson, it was, hey, durable resistance is you take a cultivar, you plant it out over many acres in areas that are highly susceptible over many years, and it's durable until it's no longer durable. Okay, so that's kind of a retrospective thing. With trees where they have to be out there for decades or hundreds of years, we don't want to plant major gene resistance like Western white pine and find out 5 years later, hey, it's toast. And so we, understanding a little bit more what is durable, that's where you can help the tree breeders like myself. Can we develop genetic markers, biomarkers that help us with rapid resistance evaluation for major gene resistance, for quantitative resistance, speed up the breeding by inserting genes in some cases, using new genes in some cases, as we're talking about chestnut. Really understanding, I have no idea what the genes for white pine blister rust resistance in our species are. Are they comparable at all to the Eurasian species? We have a trial right now where we have Pinus siberica and it has 95% survival. And some of our white bark pine have 70, 80% survival. Are they even using the same genes, mechanisms, and whatever? Be interested to know. If so, then maybe it will give us a hint. Well, hey, maybe it is going to be durable because Siberica seems to be doing okay. If they're different genes, then are they going to be durable? We heard something about speeding up flowering in chestnut. If we could speed up flowering in things like sugar pine, that would be very helpful. And all these tools need to be inexpensive to use if we're actually going to use them to solve the problems for society and keep these trees out there. Oh, okay. One other thing, I was going to talk about Sentinel trial, a project that I started with Jeff Williams and Beth LeBeau of International Programs about a year ago in August. And we want to be proactive in looking what are the next threats to, and this group is very important, the next threats to North American species. And I was talking to Beth and I sent her a short proposal a year or two ago and whatever. I said, hey, geneticists, they've been planting trees around the world, either in provenance trials, progeny tests, operational planting, Doug fir, loblolly pine, radiata pine, various other species for 100 years or more. And so there's a lot of data on there as sentinels on a lot of our major species, and with botanic gardens, even some of our minor species. Can we gather that in and develop stuff? White pine blisterus in Germany, they actually have higher resistance than some of they collect seed off of some of their native surviving material, tested against our material from North America. And hey, they get like 30% survival in the test versus 3% survival or something like that. And so, something's developed there. China, there's probably a second species of blisterus. Can we answer the question, is our resistant material going to hold up if we get that in here? For the redwoods, in China recently, there was a paper published on Metasequoia and its susceptibility to a particular Phytophthora species. I tried contacting the authors just very recently to ask the question, are you still doing work in this? Is there a chance that we could maybe work with you on the redwoods? Because it'd be nice to know proactively on this. And then also to know, I'm working with some people in Scotland to proactively test Phytophthora ostracedri against Porterford cedar, Alaskan yellow cedar, maybe one or two other cedars, because it affects juniperus over there, and it has affected some yellow cedar and whatever too. So how can we in the genetics group link with our colleagues overseas to kind of predict the future, and also whether there is some resistance out there that, or whether we're going to have to put up greater barriers to keep something out? Okay, just, any of these programs take a lot of cooperators. We have a lot of cooperators, private companies, 5 or 6 different tribes, states, counties, federal agencies, including national parks, et cetera, like that too. But a lot of people in research groups as well too. We have a ton of material, and if you're interested in well-defined material that could be used for resistance studies, we have a lot of, I think, good material. But it's not our position and job to go ahead and answer a lot of the fundamental questions that could be or should be answered on that. Thank you.
Thank you, Mr. President. Actually, set the stage for this panel conversation, I think. Yeah. So, any question in the room? Yeah, Justin.
I just had a quick question. So, you had a comment or a slide on biotechnology. So, I guess your conclusion is that you don't need biotechnology in breeding programs, and then you seem to be. You also say that resistance exists in every species. So, I don't know if I agree with that, but I guess what are your views on CRISPR and the potential for integrating CRISPR in breeding programs? A lot of agricultural crops are integrating CRISPR now into breeding programs to accelerate selection. What do you have any thoughts on that before itself?
And actually, it was much new for this year. So, hopefully, the biotechnology role and perhaps, as we talk about humans now, that CRISPR is coming, so it means that people have more access to natural variation.
Yeah, biotech, again, I think you got to separate crops from trees. And even with CRISPR and some of these new techniques, many times we're dealing with a single gene. And I didn't focus on it, but there was a slide there with Porterford cedar showing major gene resistance and quantitative partial resistance and stuff like that too. And I think if you spend too much time focusing on one gene, unless you happen to be in a situation where sometimes one gene is durable and has lasted 50 years even in the crops and stuff like that too. And I haven't seen anybody that's really used CRISPR for a quantitative trait yet. It may get there at some point in various other things too. But I think when we think of trees, don't think of necessarily crops and cultivars. And think of durability of resistance, think of stability of resistance. I think in our blisterus system, a 2 or 3 degree temperature difference sways in our white pine some of the stability of the resistance and whatever like that too. So we have to be careful oversimplifying things and stuff like that too. And I'll put the gauntlet down there. Yeah, if you have a tool and you think it can really be developed. But as I mentioned, major gene resistance is our new susceptible in western white pine, some of the fill sites that we have. And so I would hate to have seen somebody do CRISPR for major gene and go out there and 10 years later you have a bunch of failures and stuff like that too. So at least if you're going to do it, put it out in the field and maybe have some expectation why it's going to be more durable, like maybe the wheat gene and chestnut.
So thank you. I would like to invite the guest speaker of this morning to join us for the panel of discussion. And I would like to ask, I don't see him, Jason Holiday. I don't think Jason is here. Yeah, he's there. So I asked Jason to be the moderator or the animator of this. So we wanted to take advantage of your presence to ask you a number of questions, but also to open the floor to the people. And I think that I will let Jason introduce himself and start the conversation.
Why don't you give it to them and then, or can they do it simultaneously, or they can pass it around and I can stand here? Or do you want me to sit there? I'm just going to stand here because I'm like the presiding over the. Natalie just asked me to do this last night, so I'm not very prepared. She asked me 2 months ago, but I'm still not very prepared. Okay, so I'd like everyone in the audience to sort of think about general questions that address sort of forest health and things that you would be interested in hearing the speakers talk about in a few minutes. I'll start with a few questions of my own. And I think that one of the things I wanted to pick up on before I get into some more general stuff, at the end of Richard's talk, I was interested in your position that most trees species have resistance. I assume you mean resistance to non-native pathogens, to native pathogens, pests, and so on. And I haven't asked my question yet. I think that, so my question is, to what extent do you think this fairly rare resistance is environmentally dependent, and how durable is it going to be across a lot of different environments where a widespread native species might be planted in the future on the basis of breeding programs, for example, from what Jennifer is doing, or Jared, or Janice for that matter.
Each species you got to treat differently. I've worked a little bit with tan oak, and I was originally, when they produced the screening in California, helped set up a program where white paper and tan oaks. Some of the slides I talked about how to set up a program went into that white paper and everything like that. Each species is going to be different, and you have to go into it in a neutral fashion. I've seen some researchers, much smarter than I am, they go in trying to prove a point. Whereas I go in there, I'm going to test. If I see something, I'm going to retest it. Then I'm going to put it out in the field. I had some colleagues when I started with Koch in Hawaii, and they inoculate these things when they're like 2 days old. And very, very small things. And then, like 30 days later, they have results and whatever like that. And I said, okay, I'm a little skeptical. How do I know that's not very big seedlings? How do I know that's not tapping off or very soft? So, you got to put it out in the field. You got to show it's there. You got to show it's durable and whatever like that too. And so you have to be very thorough with the approach. And we all have to you have to play double. You should be your own double advocate. And my material is, I just got a note from Nick up in British Columbia. He pointed out white pine for just tabulated 7-year results and said, hey, you guys have some good results there. You got some good material. So here's an independent group up in British Columbia looking at some of the material out there. And it's only 7-year results, so I'll be happier when it's even further along in order too. But yeah. So each species is different. Going into it, you really don't know. White bark pine was a huge surprise, both to me and the other people working with white bark pine, the level of resistance, but it also varies by population. And so going into it, just like with the ashes, we sent some material over to Jennifer. I'm interested to see a small number of families, but is she going to find any resistance in our initial families of Oregon ash? Because there's people now in Corvallis, Oregon ash is real important in parts of the West Coast. For a lot of the urban areas. And I went to a forum 2 years ago, and one of the city managers saying, hey, we're not gonna plant it anymore, we're gonna look for an exotic species we can plant. So right at that point, I said, okay, I don't know, but let's see if I get a few dollars from Washington office, get up with Jennifer and see if we can look into this before giving up on it. So.
Jennifer? What's your perspective on this topic?
Whether or not resistance exists, that part?
Yeah, and to what extent it's environmentally dependent, and to what extent I'm not criticizing your work at all, but I'm thinking about chestnut, and you know, we don't go and inoculate those large surviving trees because we could kill them. And so, you know.
Let me, one other thing I want to get correct, but environmental dependent, our western white pine are major gene resistance. In some years, we get like classical ratios. In other years at Drano, we don't. And sometimes in the field, we get 10 or 15% of these families infected when they should get no infection at all. And in the crop literature, there's stuff there, especially with rust, but some other pathogens that sometimes like a 2-degree difference warmer can neutralize that resistance. So one of the questions I want to ask is, will our resistance hold up at multiple sites in different environments, or what are the limitations of any of the resistance? And I think we all have to ask, you know, there's stability and there's durability. A lot of people talk about the durability question, but the stability over environments is very important.
And I agree. I think we all know that phenotype is, you know, genotype by environment. So we expect there to be environmental influences, and that's an important piece of solving these problems and keeping aware of that breeding program. Just to address the question of, does resistance always exist? In addition to Richard's example of Port Cedar, Karen Pike was involved in a project with Eastern White Pine, where it was a similar thing, where people were not using the proper inoculation techniques. And so once that was optimized, they were able to identify resistance. And when we first started working on Elm-Bosch, everyone was saying there is no resistance. And as you can see from our results, it's a very, very subtle resistance. But once we start breeding with that subtle resistance, we get gain, and that's the important part. The other aspect of EAB is we don't ever expect to see that huge level of Elm-Bosch again, hopefully with biocontrol, keeping it limited, and as we increase resistance. So we don't expect that environment to be the same. So again, that's going to be a different type of environmental influence that would, you know, favor durability versus some of the other ones like climate change and the differences in degrees that in small differences that Richard was talking about. So I think they're important questions, but I think they're things that can be dealt with. And I would agree with Richard. I cannot think of an example where someone has looked for resistance and haven't been able to find it. I think the important thing is that people have to refine their idea of what does resistance look like, because what I faced, you know, 10, 15 years ago was this idea that if it wasn't a bulletproof tree or if it wasn't completely immune, then it was not resistant. So people lost the whole concept of partial resistance, and how having some resistance is a good thing, because through breeding you can stack these genes, you can combine, you know, different genes to help elevate the level and frequency of resistance. The last point I wanted to make too is the crop comparison versus the tree comparison. And keep in mind that the goals of forest restoration are very different than the goals of crop breeding. In crops, the goal is to get, you don't care about genetic diversity, you're looking for genetic uniformity. You want all of the crops to be as similar as possible. But when we're talking about breeding for forest restoration, we want to preserve as many diverse resistance mechanisms as possible, because that will help us with durability of resistance. And then we also need to preserve genetic diversity. We don't want all of the trees to be exactly alike, so that they're poised to be able to adapt and survive climate change and the next stress that comes along, and so forth and so on.
So is it an emergency in terms of ash to preserve that germplasm in terms of the spread of EAB, or have we pretty much reached the point where we're going to, whatever's there is probably going to persist?
Oh, no, I would say it's an emergency, especially with green ash. There's a couple of white ash populations that have been reported on where and we do find a couple of clusters of green ash, and we've been monitoring those, but And some are persisting, but some are still dying, and they become weakened and they die from, a wind event or something like that, because they're always still being attacked. And with the white ash clusters that were identified over time, what we think is happening is there was a major mortality event that made a canopy openings where the existing regenerative layer that was already there, but it wasn't of the size that was preferred, just took off and grew into these bigger size classes. So people saw these scans of a lot of these bigger size classes of white ash and thought, oh, you know what, there's so much white ash left. But what we're seeing is the AV population is slowly starting to build back up, which takes like 10, 15 years. And I think we are poised at the second round of mortality. So that's a concern. And knowing when to make these selections to get the best bang for your buck after AV has gone through and devastated things. And optimization of the bioassay in correlation to the field performance is really going to be critically important in that as well.
I was curious to hear.
Yeah.
Can you speak in the microphone?
I can speak in the microphone. Yeah, is that better? How about it, Jared? Is there always genetic resistance?
So with the American chestnut example, the large surviving trees that we've seen, we do have progeny where we cross the trees together and then inoculate them with chestnut blight, and they survive for like 15 years, and they have superficial danger, so the resistance seems to be heritable, but the base of diversity is very low, it seems like. So I think like having these other tools has helped, like having the back cross breeding to bring in resistance with Chinese chestnut, and having a transgenic approach has helped us expand that base of diversity compared to just using the natural resistance.
Janice, do you have a comment on this topic?
When do I not have a comment? So I would say that I applaud the idea that we need to have an open notion of what resistance is, and certainly with the pines that we have identified, by we I mean staff has identified on landscape of survivors, they are not resistant in the true sense, but they were resilient in that attack. So I think we have to have an open mind on how we define what that resistance comes to. So that's to support Jennifer's comment. And I think we do need to take environment into account. One of the other systems that we work on is Western Walrus, where we see less attack at higher elevations, but those trees aren't necessarily more resistant. It's a disconnect in the phenology of the sporulation and the bud burst of those trees. So we do need to take environment into account, and particularly if we're thinking about things like drought, that's going to certainly affect those trees' interactions with our insects. It's going to affect their interactions with our pathogens, but particularly our insects. That's where we can see the biggest impacts of those environmental factors on those interactions. So if we're not considering environment, I think we do have a problem. The other thing that I would like to point out is that some of the identifications of resistances and breeding may not have application over wide forested landscapes. We may not have enough genetic diversity. We may not have the right material for deployment in those regions. But where it can make a difference is in our urban landscapes. And I think we need to be thinking more and more about making sure that we can keep trees on the landscape in urban settings. Living in Edmonton, our boulevard trees are American elms, green ash, white ash. Do we have a problem coming our way? Yes, we do have a problem coming our way. Emerald ash borer has already been through Winnipeg. It is already in Regina. Is it heading our way? Yes, it is. I have a green ash in my front yard. It's already taken a hit from other sources. So is it a goner? Probably. But that's a source of genetic diversity that hasn't been plumbed, are the areas where these urban trees have been planted in areas outside of the native range that maybe we can take advantage of.
So stepping back a little bit to a more general, just general notion of forest health. And if we're out on the beach and we see this wave coming in and it's sort of green and then it crashes, right? Where are we at in terms of our risks to our forests from both native pathogens and pests that are interacting with climate change and non-native pests being introduced? Are we sort of seen most of what's going to happen, or are we still sort of going blind and don't know what's on the horizon and what's going to be the next ash or the next chestnut?
There's new, and the entomologists and pathologists have documented this. There's new pathogens or insects coming in all the time. And so there's a real fear. There's sentinel trials being set up by different countries around the world. Everybody is worried about this. What is the next threat? And so I think it will happen. There will be new ones. There'll be a new interlash, or there'll be a new blisterus, or phytophthora, things every week they come up with a new phytophthora species or hybrids of phytophthora and whatever like that too. And if you get ones like Cinnamomai, which affect 5,000 plants or species, like was mentioned, that's really scary. And so I think we have now reached stable state. Climate change expands that. Cinnamomai is moving north with the warmer, likes warmer soil. So as you get warmer, some of these pathogens are going to, get into a new range. And, they may become more aggressive. What are the optimum temperature ranges? I was working with some people in Hawaii on Ohia, and they were having problems with their inoculation. I said, well, what temperature range is this pathogen? Because they had a greenhouse and an outdoor, and they were working in the greenhouse. What temperature range is this thing optimum at? And, they told me that their greenhouse was a few degrees off of that, whatever like that too. So
if that's the case, I think probably everyone would probably agree with what Richard just said, that we've got a lot of stuff out there that we don't necessarily know yet even. But how do we prioritize the species that we respond to? Because we're not a huge group. I mean, it's obviously this isn't all the people in the world that are working on forest health, genetics and breeding and so on. But how do we prioritize the species that you invest the amount of energy that, say, Jennifer is investing into ash? Maybe Jennifer can.
That's ultimately up to society. Dina and I remember, maybe somebody else in this group remember a group called P4, and so they're interested in the 4 southern pond species. And right now things are kind of okay, but they're saying it's just a matter of time before something comes in and moves those. And so they want to develop a plan to be kind of aware, proactive. If it comes in, how do we alert people? How do we immediately look for resistance and whatever like that too? So, it's coming. When it will come, who knows? And
the one example that came to my mind was in the east. We have beech hemlock forests, and we're seeing more of the chestnut. We now have 2 different species invasive threatening that species. But with hemlock, we have hemlock woolly delta, hemlock scale with beech bark disease, and now we have the new beech leaf disease. So, and then on top of that, climate change. So to me, do we want to lose an entire forest type? And then if we do, what's going to replace it? So those are factors that go into prioritization. The other thing that just popped in mind with Richard talking about that, how to get people to mobilize and think about resistance even quicker and sooner is, I think the proactive studies that I was trying to talk about, where there's areas where black ash has not, EAB hasn't reached the majority of the northern wetland forests, where 300 million acres of black ash could be converted to non-forest type. We have a chance to actually, put resources and start planting improved seedlings before EAB gets there, so that we can, hope that regeneration has some resistance in it. And it's the same thing we're doing with Oregon ash with Richard. So that's another way to sort of be more proactive, and then also get people's attention on maybe we need to look for resistance sooner rather than later, and not rely on an insect to kill 99% of the trees before we say, hey, that one's still alive, and because we may have lost too much of the genetic diversity like we have with chestnut. That may be the case with green ash. You know, we may not be able to get resistance to the level that we really actually need. The field test will show we may not have the diversity left, to your earlier question. So, those are my thoughts on that.
Okay. I think the economic species will certainly come first, but beyond those, you do have some like maybe green ash, white bark pine. We have a group from Sweden, and they'll visit them for a couple of days, and they're coming to this winter. Scotch pine, the rust suddenly is a native rust, but it's suddenly on the uptake, and they're really worried there too. So they're going to come out and they want to potentially set up a resistance screening operation, but they also want to know from a site hazard and various other things, what can they do? And I think climate change is maybe partly the driver of this. So both for our natural pathogens or insects, as well as new ones, you know, the first going to be out there. And as I told people in Hawaii, we don't have time to reinvent the wheel. So whatever we can find successes and people can, you know, like, you know, there's no way I was going to start an ash streaming program for various reasons. One, it's not in Oregon yet, so I don't want to be the one that accidentally introduce it out there. But also, you know, using existing expertise elsewhere and stuff like that. And I think we've got to do more of that.
Jared or Janice, do you have anything to add there?
I think, just like COVID, you never know when it's going to hit and when it's going to hit. And I think as a community, we need to do our very, very best to get these problems onto the radar of the people who have the potential to enact policy, to enact practice really, really soon. And, you know, it's great when you can have miles of dead red forest draw attention, or the ash in urban landscape draw attention to this and get the public backing. But by then, as we've heard, it's an uphill battle to make a difference. So how do we as a community get the attention of policymakers earlier? And I'll use the example of mountain pine beetle moving into jack pine as an example, where the provincial entomologist in the province of Saskatchewan was trying to get his minister's attention to do early monitoring for mountain pine beetle, and the guy was like, don't even talk to me until it's in jack pine. I don't have a problem because it's not going to go into jack pine. And then when the paper was published that morning at 9 a.m., Rory got a call from his minister and said, how big do you want that check? It took something pretty dramatic, but it took definite evidence, scientific evidence to change that. And that actually has brought about a 15-year collaboration to make control a reality for Eastern spread. But how do we do that sooner? And why does it have to take a calamity? So, Dave, I think you're leading a discussion group on advocacy. Maybe that's something that we need to be doing more of, is trying to get the ear of people who can make a difference at policy level sooner with scientific credibility.
One of the things that's worked for the American Chestnut Foundation, it's been a liability and strength for us, is involvement of non-scientists in the process and them being the advocates for us. And of course, that is like herding cats too. But people pay attention to the story, and we have a lot of people that will like say, like, I have this tree, and like, you know, is it an American chestnut? And so we have like this network of people that's helped us out a lot. And I think that's been a liability for, I guess, the political side of things, like getting the transgenic tree through the regulatory process, knowing that there's a group of citizens that really care about this, like, has been useful for the government agencies to see, like, this is important, we should make this a priority. I don't know if that's happening with ash or anything, but I think that's like something that would be really helpful too.
Yeah, we are getting, well, first of all, as federal employees, we can't really be the ones to speak directly to policymakers. So, so it's good that you're encouraging others to do so, because I cannot. But anyway, we are doing a lot more work with outreach because what started happening with our beach program and with ASH is that I was constantly getting emails from people saying, when can I get resistant seedlings? You know, when we are just getting started. So it's really obvious there was a lot of interest among people. So I didn't get much time to go through it, but some of the organizations that we've been able to get some funding for and get started are focused on sort of extension and tech transfer and recruiting all sorts of partners at all levels. Partnering with Tree Snap and the other one is that ashmonitoring.org, where we're getting people that will, there's groups of people, volunteer organizations that work with parks. So what we encourage is what we call train the trainer. So we always want a natural resources professional on the ground to oversee the groups of volunteers. You can get volunteers that want to install plots and they'll collect data annually. And at this point, the Ecological Research Institute has identified their first 22 lingering ash as a result of 60-some partnerships in the state of New York. So I think getting people involved is a really good thing because they do get excited about it. And then the follow-up is really important. The American Chestnut Foundation has done a really good job with the journal and the outreach you have to your members. But that's something that's more difficult for us to keep up with, but people want to know that their efforts are actually getting somewhere. Partnering with states was really important too, at least here, because the states have become more and more vocal up the chain, I guess, is the way of saying it. I think that was it for our outreach, but I think it is really important.
I think one of the keys, a lot of us when we're in grad school, we do your research, you move on, you kind of keep going there. And we weren't really told to get out the news of success stories. And so I think when you have success stories, we need to get that word out now. We're supervised, we're a regional unit, but I'm located on the International Force. So technically, the force supervisor of International Force is, head of our unit kind of. And many years ago, when there was a previous one, he asked for a regional review at some point of the whole force. And he said, now's not the time to be shy. And it's to get the word out. And he asked for an escalated review out of the rotation of his force and whatever like that too. Because you want to show what you're doing for the taxpayers' money and for the public and whatever like that too. And I think many of us weren't schooled with that vein. And I think if we don't, we'll go extinct. I see the ARS newsletters all the time. They're always talking about, they do a pretty good work at getting things out and showing the cost benefit and whatever like that too. But us as forestry, we're kind of the best kept secret type thing. If we do have success, you just need to get it out. And in genetic, climate change, this could be a huge factor. There's very little that management can probably do. It's going to come down to species choice and genetics and some of the new genetic tools and everything like that too. And certainly enlisting any of the members of the public that we can as well too. But so I'm real excited to see a group like this start, and I'm hoping it increases the visibility. In Congress, it's stuck in there, but it's got co-sponsors, and it's got groups like SAF, Society of American Foresters, the state foresters endorsing it. But there's a bill in the House that would boost activity and funding for forest health, and part of that was to increase some of the genetic components and stuff like that too. We'll see if that ever makes it through. But making that type of thing known so that other groups like the state foresters, Society of American Foresters, if people believe in it, can make it known, and hey, maybe it will rise to the top and funding will come out for where needs actually produce. You can review our program every 3 years, every 5 years. Hopefully it's moving forward. If it's not, then you should, change something about it and stuff like that too. So we shouldn't be afraid of reviews, but we need to get success stories out.
Yeah, it would be great to see if this organization can help with that aspect of getting out success stories and communicating a lot about the value of genetics and breeding and the genomic tools and how they can actually help with these processes. Because as scientists, like Richard said, that's really not most of our forte. That's not our strong point. And it's really hard because it's hard to keep up with everything. Like my program, for example, I have my salary covered, I have facilities, and I have one permanent technician up until right around the time the pandemic started, I have a second permanent employee. Everything else has been funded on soft funds, and that's a full-time job just in and of itself. So it just becomes sort of this insurmountable at first. So any organization that can help escalate the whole conversation and the whole visibility of breeding would be, I think, really big added plus.
Do you have one brief comment on this, or are you done?
I think that's the
No.
question.
Yeah, I wanted to allow the opportunity for the audience to jump in, because we only got about 10 minutes left. Andy?
Yeah, so public outreach and that sort of stuff is obviously really critical. But in listening to all the talks, like Richard made the point that, I can get you materials, but I don't know why they work. And Jennifer is on kind of on the other side, like materials, or you're figuring out why they work, but a very small genetic base. So, in addition to the public outreach, I think there's a increasing that overlap between the applied management and the research is that almost seems to me like that has to happen first, or in addition to the public outreach. And maybe that's something that the society can also help to to increase the overlap in that Venn diagram.
How did outreach in TACF, how did the two sort of, did it start out as a kind of joint effort with lots of citizen scientists, or did it kind of like come in after?
Yeah, we had like a research farm, and we were like literally operating on someone's like house in West Virginia or something, and it was like really small for like 20 years. And then we had like, a base of material, and then we started reaching out to people, and they knew where the trees were, and they did some breeding. They contacted their utility companies. It was like really grassroots for a long time, and that's actually, living with that is like, it's been challenging because we have like orchards like all over the place, like just to get to all of them, it's taken me my entire time at TACF since 2015 to kind of see the entire thing. So it's hard to manage. And one of the things that's opened up is our ability to do fundraising, so people know about us, and our ability to like go and get private funding. So like we have our staff, we have like a whole just branch of people that are meeting with like rich people and like getting, private funding for a lot of this project and that project. And we've been kind of cobbling together enough to, do this genomic sequencing. It's like not federal funding. It's like somebody gave us a gift, and we went to JGI and they did it for us. So that's like that kind of non-traditional, like non-federal funding has been really propped us up, and that's really because of all of that outreach we've done has opened up that pathway. So I don't know. With the ASH program, you had like multiple orchards that are working on lingering ash or something like that, and you establish that. Like, I think that could be a big asset to the thing, and then having a whole fundraiser. You're a government employee, so.
Actually did the private funding recently.
Yeah.
But we can't solicit it.
You can't solicit it, yeah.
They came to us.
I just wonder about this like public-private setup, like where you can have an ASH foundation or something like that feeds in, because we do fund forester screening and things like that too. So we wouldn't been able to do that through. Like I don't do all. Obviously I'm not doing all the work. We have to collaborate with scientists. So I'm just kind of like in the middle of all these collaborations, but mostly other people do the work. And that's been really important.
Andy makes a good point. Is plant breeding has been around 1,000 years, a very effective thing. And I made the point when I was over in Hawaii talking to Bahia, I was asked to give a talk there on the Bahia Depth, and they said, don't let this program get stuck in research. Okay? Full speed ahead with the breeding. work with the researchers to answer some of the questions, but if you wait until you have the answers to all the questions, the species will be functionally extinct and stuff like that too. You know, plant breeding is a very, Norman Borlaug, Nobel Peace Prize for Green Revolution. I saw an American Experience thing 5 or 10 years ago. He was down in Mexico, planted 100,000 corn plants at the end of the season. I think he had 4 left or whatever like that. Just a number of them. He didn't worry about answering, well, how many genes are behind it and various other things. It's plow ahead, show success, then because sooner or later, the public's going to ask, okay, hey, we've been funding you for 20, 30, 40 years. You haven't planted one tree out there in the forest yet. When is it coming? And whereas I'd rather say, hey, can you tell us we're carrying along all the QTLs and various other things? So, it's great to have money both for the basic research, but also for the applied. But, don't slow down the applied part to answer all the fundamental questions. Because like I said, listeners, I can't tell you how many genes there are.
Janice,
I
what,
can't change the underlying mechanism, but I can tell you, it's working. We have private companies, tribes, men orchards, BLM, Forest Service, National Parks is using the material. So far, people are happy with it. I would love for researchers now to interact with us and find out, how many genes we do have and whether the different major gene resistances are different and whatever like that too. So, same thing with assisted migration or climate change eventually too. The same question. If we wait till we understand every single little nuance, we won't have any trees left.
Janice, what's your perspective coming from a place where the government mostly manages the forest lands and planting and deploying potentially your resistant material or the results at least that might lead to resistant material?
Yeah, we're on a step 1 of the 12-step program, I think. We've been able to establish that there's genetic differences. We are pretty far away from deployment. And particularly, the government of Alberta is surprisingly conservative when it comes to deploying genetic material on the landscape relative to other jurisdictions. They're pretty kamikaze when it comes to any other environmental thing. But really pretty conservative when it comes to the genetic side of things. So we have to be able to show a great deal of promise and really that on the ground truthing before it would ever be picked up in Alberta. I think we have a stronger opportunity in BC where there's already a tradition of genomic selection and of select seed. That having been said, it's one trait and there's lots of threats to our forests. And so I think this is where as a more fundamental scientist, I throw the ball back to the breeders to say, how are you going to hedge for all of these different traits on the landscape in a changing environment where it's not going to look the same in a century, let alone further than that? It's some pretty serious calculus. And so that whole bringing to management and deployment is pretty tricky.
Question, Brad? No. Sally. Oh, Sally had it. Arm wrestle.
I have lots of questions, actually.
It's one question, Brad.
I just want to put it in plug for common garden study. And I'm glad that you guys are talking about environment and that, resistance is gray area maybe because of the environment interaction. And an example is we had a journalist visit one of our study sites, reciprocal transplant study. And the first line of her article said, I drove to Oregon because I wanted to see the future. So and this has to do with climate change. And what she saw, she called arboreal apocalypse. And what's happening there at this site is trees moved from a milder climate to a hot, dry climate are getting attacked by Thermophilus, followed by those for bark beetle. And we wouldn't know, nobody suspects this until we start to move things around a little bit and see what happens from warm, from cold to warmer sites to look at climate change, or from warmer to colder sites to look at the effects of the system. So my point, I have lots of questions, but my point is we need to also be looking, remember common environment study.
Agreed. Since we're close to the lunchtime, maybe I'll just let Sally make, if she has a point or a question.
Yeah, I have a question that might also be a point, but so you all talk about genetic diversity, genetic base. I know when you first have a disease come in or an insect come in, you're scrambling for those first few individuals time. But what I'd like to know is if you are setting targets for deployment on diversity and how those targets are set, what are the parameters for that? And are you comfortable deploying before you get to those? So, and Derek, I know you've done a lot of things about this in the last few years. You might want to wait 10 days on your talk show. You might want to start that very slowly.
Well, that was exactly what I wanted to talk to you about, Sally.
Me too.
And so like, one of the things we're doing is like whole genome sequencing, Jason will talk about it, whole genome sequencing of a range-wide collection to characterize the climate adapted diversity. And then we're sequencing the background trees that we have, and we're seeing what is the adaptive allele content of the stuff that we've already conserved. And then what I don't know, and I'd like to figure out, and I don't know if anyone in this room knows, is there a test of like sufficiency? Like we have adequately not only captured, like from an SSR point of view, like the rare, I mean, the alleles, 5 alleles or something, but like the total adaptive content of each of the subpopulations. And are we talking about individual alleles? Are we talking about a combination of alleles and climate adaptive envelope, maybe, relative to what is projected for climate change? I mean, I think that's kind of the direction that we're thinking, but I don't have the specific like statistical tests and things that to figure that out. But we're generating baseline data to do that, and kind of developing those metrics, I think, is an important thing. Maybe you've thought about this more, and that's what I want to talk to you about.
I think that one more response and then we'll have to head for lunch. So Jennifer, do you want to?
You are right. We do have to scramble because we have to prove that resistance exists. So we have to work with what we got, but now we're very aware that the clock is ticking. And although I agree to a certain extent to Richard's point that we can breed resistance and we don't need genomics to do that, to get it to the pace and scale to restore an entire species or to actually prevent forest conversion that I was talking about, I think we do need more modern tools. And so that's one of the things that the funding that we were able to get in our partnership with TNC is going to help us accomplish, is to do some range-wide genetic diversity analyses on different ash species, some in conjunction with Canada, using provenance trials that are left surviving, seed collections, and things like that. But it's definitely a challenge, but unfortunately, I spent 10 years proving that resistance was a thing before we even had the luxury of thinking further than that. And to the point of the common garden studies and even reaching out to management is that if we have the funding to really do breeding at the level that we want, we would have plenty of stuff to be putting out in common garden because we would love to do that. And I agree with the managers. I think the key is reaching out to management, and we have tried to do that. And I don't know how we got the attention of this particular funding source of funding, but let's just hope it is a trend.
You have an email address that the federal government isn't aware of and sent them an email.
No, actually, they have been funding American Elm Breeding Program, and it's a foundation where they grew up in New England and they noticed changes in their forests, and they wanted to address specific species because of things that they've noticed from changing throughout their lifetimes. So it's kind of cool, but it's also, it's not something that you apply for. It's something that they come to you and say, we want to fund this. But I know there are places that people can apply for funds through philanthropic agencies and things like that. So, we used to always joke about that, that we need our own, nonprofit.
Yeah, definitely.
To do that.
Well, I think we're gonna have to wrap this up because lunch does not wait for us, and it'll close at 1 o'clock if we're not there. So let's thank our speakers again and for this good discussion. If you have additional questions, please corner them over lunch or throughout the rest of the meeting.
If the moderators for the next 2 sessions can just come up here really briefly. Oh, I got it. That's right, 120. 120. Yeah, you're first. You got it. Woo. All right. Thank you. How's it going? Good. Enjoying it. I know, it's really great. So I just want to make sure you know what you're doing. So you're going to be here for the afternoon session, and you have 2 talks. I think he posted both his PDF and the PowerPoint. One thing I'm not sure is if we have audio working right now. So I'm going to try to get that working because we had some issues with that earlier. And then I just want to make sure who else. You have a 3rd talk in your session. I thought there were a couple. I'm just gonna make sure you have everybody you should have. Emily, Jason. Okay. Let's see, they might actually just be in the. Emily here. Emily. I just saw her, but I think she wanted. So okay. Google document that there. His is there, but Jason is there. So you have everybody for your sessions. I'll just make sure it's syncing properly. But you can pull the all right, cool. Video will come up to the talks. So these are once you load it to who's first? What am I? Right, so let me just load his. Hi. Okay. That's not a problem. Well, we can check it too, just to make sure it loads okay. And you're this evening? You're not this afternoon. Yeah. Oh, you're tomorrow. Okay, we'll check it later today. Just make sure it's running okay. And then we'll make sure. And then so we can play it from here also. Or for his, we can download it since it's in Google Drive. Hello. Did you figure it out? No, not yet. I'm gonna check the audio because I think he might have. We couldn't get it to do that, but his is there. So okay, does that work? But I'll try the audio again to see if we can figure it out.
Yeah, so in this schedule, lunch is over at 1.
They will not serve past 1.
I know, but we don't start until 1:20.
That's right. Yeah.
If you heard people like a few minutes early, Beth would have a few more minutes to talk.
That's true. I think we're actually going to be okay because I think the person in her. Oh, Beth wanted like a couple more minutes, which is fine. Okay. Because it's fine. Because that one can be shorter. so if she goes over like a minute, don't hurt her off. How do you want me to introduce people? Because I don't really know people. So we're only doing the long bios for the invited speakers that like Natalie did. But these guys, it's just name and institution, and you can list the title. It's listed.
Oh man.
Sorry, the
University of New Joaquin de San Nicolas.
He just came by. He was the one who just came by to ask because he was first. So if you wanted to, you could also Hidalgo. University.
I talked to Laura and she told me how to pronounce his name. So.
Okay.
Guatemala.
That's right. Yeah. So that is going to be his first. But let me see if I can get audio working really quick. And the other thing I have to do, I need to track that down because we don't have access to the breakout rooms yet because the keys don't work. So
oh, it's fun.
Yeah, so we'll do that next. But You're all set. Well, before you get there. It is so hard. So, yeah, I got the key, I think, so I'm gonna try the rooms, and see if that works. Like I said, if that goes, then we're good. I was gonna text the crypt to the other two rooms. I can see. Check the keys. Morning was fine. It went really well. Yeah, I thought that was great. Yeah, that was perfect. It was a really good discussion. So, anyway, so we'll come here, then Dave's gonna do his little presentation, and then we are going to, I'll put the versions up early. Okay, I'll do all that. So I will be with Kathy for the lunch. Oh, great, okay. So we will just have a chat. Perfect. Sounds good. I'll text you guys if you need anything. Okay.
So in the morning, we were trying to play the Canon video, but
Okay.
for some reason we thought it came from the projector screen or it didn't come from the normal speakers.
Yeah, it should be playing, if we set it up earlier, so just go to system preference.
Yeah.
Okay, so just go to your, where's the speaker
I see sound there.
at? Okay, right there. So you go to output,
Ah.
and right now selecting external speakers, which is this right here.
Okay, so essentially now it should probably be working. Okay.
So was it this one?
Yeah.
Do you want to play it so
Yeah.
we can hear it?
Yeah. Oh, wow. Okay. Anyways, yeah.
It could have been that the volume was down,
Could have been.
so that's probably.
Yeah. Okay. Well, it's working now, and I know how to fix it if it doesn't. But
Okay.
thank you. Thank you so
You're
much
welcome.
for making your
No
trip.
problem. No problem. I'm gonna be stepping out for a few minutes,
Mhm.
and that's about it. I won't be back probably for about a good hour.
Yeah, I think
so?
the next talk is on like the recorded talk is only in the evening. So
Okay.
it'll be fine.
Okay. If you can just do me a favor at the end of the day, turn off the projector. There's an on and off button.
Mhm.
You just push it twice.
Okay.
It will push it once. It will display. Are you sure?
Yeah.
You just push it one more time and it'll turn it off.
Okay. At the end of the day. Sure.
Yeah.
I'll do it.
Okay.
Yeah.
Thank you.
Thank you so much.
I will be here in the evening, but I won't stay here throughout the whole entire.
Yeah. Okay.
Okay. Well, thank you.
Thank you so much.
And your name was
It's Vidya.
Vidya?
Yeah, Vidya.
Nice to meet you, Vidya. Okay.
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 Yeah.
Thank you
so much.
And how's the microphone?
That worked perfectly this time.
Okay. No changes?
No changes.
I should check the batteries.
I
Thank
think
you.
it'll last today. So.
Yeah. Okay, thank you, Vidya.
See ya. Thank you. I just feel like I'm just clicking buttons at the right time.
Yeah, the button's right here. So it's this button right here, the big one, you just hit it once, and you see how it goes up, you just hit it one more time. And then slowly. Oh, thank you. Have a good one.
Yeah. I'll try my best.
Who is controlling the time? It doesn't work. So how I need to,
So you want to try to point?
so I want to point here, but
Okay.
how can I change?
Oh, just with the arrow. Right
Arrow
here.
key, okay.
Yeah, so if you try that.
Wait,
Oop, hold on.
about this pointer option.
where is the person?
It works.
That one.
Now
That was
should
his, but it doesn't
Okay.
look like it's gonna, it's the one thing.
Everybody
can get mad at me if I don't.
That's
right.
This is my microphone.
I could try
Like,
adding
the
it.
mouse will work if you want to
Yeah.
try to point to.
Okay, I'll
Yeah.
try that.
But it's the one thing where you have to drag it to the other screen, right? So.
And I-
Oh, okay. So that messes you up.
I like to walk,
Maybe I'll
I would
try
like to
this
walk.
game.
Can I
Yeah,
use this
because I
one?
can't. I have a bad habit of turning.
Yeah, I
to
think
the
that should work.
thing and then- Yeah. Everybody watches my back. Yeah, I'll try that. Yeah. Oh, for
Maybe
this,
to show- yeah.
do you want to walk or did you want to?
I think I would like to walk, but if I walk, I cannot change that.
Exactly,
Well, okay,
yeah.
then I will stay.
Okay, sorry.
Could I,
Oh, you have it.
I just want to try if this
Sure.
is on. This works?
Absolutely. How to do that?
Yeah, so just click the,
Okay, then I want this one.
So we got it to work?
yes.
Okay. Let me get it. No.
And the time.
Oh, the time is.
So you want him to give you a signal? You can ask him for a signal. But he's generally.
Yeah, so it's 1 hour.
That's myself here.
All right, everyone, it is time Come on in. Yeah, that's what we'll do.
And they will not be wrong.
Yeah, no, we will. We'll have a coffee.
All right. Good afternoon, everyone. We're going to start with our afternoon talks. First of all, I'll just thank you to the organizers organizing this and making this happen. This is a great place for it. It's personally my first time in the Silmar, so love it here. Thank you so much. Okay, so Cuauhtémoc Sánchez Romero from the University of Michoacán is going to tell us about a fur that's endemic to Mexico and some population dynamics with it. So,
So, Aries religiosa, sacred fir, is the what creative forest where overwintering the monarch butterflies that migrate from northeast United States to Mexico. Is the same species, but not the same population that the ones that come very near of here at the sanctuary that is very close. And they go to a very, very specific site in the neovolcanic axis, which is a mountain chain with very high altitude. And we have this monarch butterfly biosphere reserve, where you can see this very spectacular aggregation of monarchs. The problem is climatic change, among other problems, because inside the monarch butterfly biosphere reserve, climatic niche modeling indicate clearly that the suitable climatic habitat for the sacred fir is going to disappear completely by the end of the century, even when I think our reserve is more or less successful because ecotourism provide an income for the local people that put them away of the logging. So we have, I think, a success case of conservation, but now conservation in situ looks that it's not enough. And what are the, sorry, that was, what happened? I don't know, it's moving by itself. What are our management options? Our option will be to move from the monarch butterfly reserve to places at higher altitude like Nevado de Toluca, which is 4,700 meters. The summits here are 3,500. And we have established, and I like the comment of Brad Sinclair of the importance of the reciprocal transplants, because we put a transplant at low altitude, 2,600 meters, and we could see the future. And when see the future, I mean, we have a reference site at 3,000 meters, moving from 3,000 to 2,600 is 400 meters, which is equivalent to 2 degrees. And we could see what happened with 2 degrees, a massive mortality of the seedlings. Here is just the beginning of the experiment. What we have now, 6% of survival with 2 degrees more and 19% less precipitation than our reference site. So it's really a tragedy what may happen, and we can see that at low altitude, what will happen. We have constructed this zonification based on common garden test, measuring how many meters of altitudinal difference makes two populations genetically different in quantitative traits, based on methodology developed by Jerry Reffeld at Moscow, Idaho, who inspired in his work. And it's why we have this 350 meters of width of the altitudinal band. So our suggestion for management is collect seed here, produce seedlings in the North Sea, and move to the next one. Not only because the width is 350 meters, also because to shift the climate for 2030 at higher altitude to compensate about 1.5 degrees. It is relatively easy to say that, but to especially at high altitude is a different history because it's complicated. Go to the field and do it. The problem here is we cannot move from some tree to somewhere inside the monarch butterfly because the summits are right there. So we have to change of mountain to another volcano. This is a previous experiment of assisted migration inside the natural distribution of the species, and we found the extremely importance of use shrubs as north plants. So a plant that provides shade and decrease the variation of temperature, either high temperatures at the middle day, because at 3,000 meters there's a lot of insolation, and at night temperatures below zero. So the oscillation of the temperature decrease a lot under the shade of these shrubs. Is what we call it protected assisted migration. Migration because we're moving the provenances, assisted because require the human intervention, and protection because we need these shrubs as north plants. And the difference is huge. So we have more than 90% of survival under the shade and only 5% of survival out of the shade. So it's a very delicate stage of this species, the seedlings. So they die very easily. So we decided to go to Nevado de Toluca, which is outside of the reserve, and we established 4 provenance tests from provenances collected along an altitudinal gradient in a way that we could test how much we can move to higher altitude to expand the upper limit of the species. The upper limit of the species, 3,500 meters. And this is our reference site, 3,400. And we have one at 4,000 meters, which is the timberline. So we wanted to see in extreme situation how we'll do this species. So the preliminary results after 1 year of planting, which is the more critical year for the seedlings, is this one. So for every 1 degree that we move to colder place, we have about 5% of mortality. The mortality is kind of messy for several reasons, but it's more clear that we see a stress index, which is based on the appearance of the seedlings that we have test this system. Is very simple to do it. So green, dark color looks like healthy seedlings, and brownish, yellowish seedlings is a stress seedling, and we have an index with 6 points, and you can see how moving to a colder side, the seedlings are more stressful. This feeling that I have is that is making very stressed the seedlings is a combination of what Jerry Redfield might call a climate without contemporary analogous, meaning a climate that doesn't exist today, or it didn't exist in historical climate, meaning at night, still at 3,500 meters, 4,000 meters are very low temperatures, below zero, 5, 6, 7, 8 below 0 Celsius. But what is new, that is usual, but what is new is that winter and spring are now very dry. So the total amount of precipitation has not changed much, but the distribution of the precipitation has changed a lot. So rainy season is more violent, at night it's more violent the rain, but the dry season, cold in winter and warmer in spring, is much more dry. That makes this combination of temperatures below 0 at night and very dry make the season very special. So we are hanging on there, and we will see what happens. Thank you very much.
I think we have time for a few questions.
I really make an effort to use only 8 minutes, so I really appreciate you.
Yeah, no, you were spot on. I have a question. So how much gene flow do you think there is occurring between the bottom to the top of the mountains?
I think there is a very strong gene flow. There are measurements of that, not mine or other colleagues of human. But the pressure of the selection by the temperature is so strong that we have shown in previous common tests that there is a strong genetic differentiation in response to the mean temperature of the coldest month. And it's about 350 meters that made of the alticular difference that made two populations genetically different for quantitative traits, such as growth resistance and elongation of the shoot. So despite the gene flow, there is a lot of selection pressure due to temperature. Right.
You looked at the relationship of mean annual temperature. Did you try other variables or multiple variables to try to account for what you just described?
Not yet, but we will do that. What happens is actually we have data from 11 months, and we're waiting for more data to explore other variables. Usually, another variable that works very well to understand what is going on is the annual dryness index. So degree days squared divided by mean annual temperature. That is very powerful to understand what is going on.
Yeah, thanks. Thank you.
Thank you.
Oh, sorry. Oof, oof, oof.
Get it going again.
You want to get
Sorry that I removed it.
No worries. I should have left you.
Beth.
This one?
This one. That's the one.
Oh, that's over there.
No, no, so that's the.
She's the problem. Sorry.
Okay. There you go. Okay, so thank you. So Beth is gonna give the next talk. She's coming to us from UBC, and is gonna tell us about the Western Larch.
Thank you. And I also just want to thank the organizers. It's really nice. This is a really great reintroduction to in-person meetings again. So I am a PhD candidate in Sally Aiken's lab at the University of British Columbia, and which is located on the traditional territory of the Musqueam people. And my thesis is looking at range-wide patterns of local adaptation and adaptive potential in Western Larch. And this is part of a larger collaborative project termed co-adaptory, the following adaptory. So I don't need to convince anyone here just what a major threat climate change will be to tree populations that are historically adapted to climate, or adapted to historic climates rather. And the risk being that there's going to be a mismatch in the future with predicted climate, with future climates. And so there's a pressing need to understand the adaptive potential and the potential for mitigation strategies like assisted migration for tracking future climate change. So Western Larch is considered a strong candidate for testing assisted migration. It's actually been approved to be planted operationally outside of its range in British Columbia, limited to a small percentage of trees in a stand. And so just a little bit of background on Western Larch. Western Larch has a relatively small range and is one of the few deciduous conifers native to Western North America. It spans a relatively narrow geographic and climate gradient, and the map of the range is depicted here. Based on previous provenance trials, speaking of Jerry Reffeld, we know that there's high standing genetic variation for climate relevant phenotypic traits within populations, and there's low population structure. And so based on species distribution modeling and the old provenance trials, the models project this expansion of areas of suitable climate for Western Larch, especially in British Columbia. beyond the current range in the future. So my research aims to tackle a little more deeply the patterns of local adaptation to climate, taking a multifaceted approach of looking at genomic data, environmental data, and phenotypic data, and in particular, phenotype environment associations and genotype environment associations. Today, I'm going to focus in on work from Common Garden experiments I've established at on UBC campus in Vancouver with the phenotypic data. So my primary question is, how are Western large populations locally adapted to climate? And some components of that I'm looking at are how much population differentiation exists in climate relevant traits, how much adaptive potential exists within populations, and which aspects of climate drive population differentiation in those traits. A follow-up to this, since we have breeding programs in BC, is how selective breeding for faster growth affects climate adaptation using those same common gardens. So the sampling for this project is range wide, and I've established a common garden out at UBC here in Vancouver, outside of its current range, with 52 natural populations here depicted in the blue and purple, and 28 breeding families from 2 breeding zones in BC. I'll just mention also that we have sampled a provenance trial in Southeast BC, the yellow triangle, for genomic data, and we're using exome capture pool-seq on 40 natural populations in the red and purple. So the traits we measured, the traits we measured last season in the Common Garden experiment include height growth throughout the season, and so the number of observations are below, just to reflect how much data we've collected. And then we've measured bud phenology in terms of bud break and bud set. And then I'm also particularly interested in drought resistance, since this hasn't been a focus, at least in Western larch. And we used this dark adaptive chlorophyll fluorescence and its declines over the drought treatments, which I'll describe next, as a measure of drought resistance. And then we also looked at cold injury in the fall as a measure of cold hardiness. So for the drought treatments, we erected drought covers in early May, and we didn't water the drought treatments for about 5 months. And the control treatments were regularly watered. And you can see some of the differences here near the end of this treatments. So of all the traits measured, we found high variance within and low variance among populations. And that's visualized here with an example of annual height growth, with the green representing the within provenance variation and the black the among population variance. So VPOP for each of the traits we measured was about 8% for height growth, bud set was 11%, bud break at about 8% again, and cold injury was actually the lowest at about 3%. So for the trait environment associations, I'm going to show you the top single predictors for each of these traits. So for annual height growth, we found that provenance longitude has the strongest correlation, with the eastern provenances having the highest annual height growth. This is also reflected in greater continentality of those climates. For bud break, the strongest correlation was also with longitude, and the eastern provenances broke their bud a little later, reflecting the greater risk of late spring frost. For bud set, we found that provenance elevation had the strongest correlation, with higher elevations having later bud set, reflective of that early risk of fall frost. And then for cold injury, we found a weak association with mean coldest month temperature. But none of these single predictors predict that much variation among populations, highest being for bud set here at 16%. So for this measure of drought resistance that we use, fluorescence decline, we actually found the highest variance among populations at about 14%. We have not found, at least based on single predictors, any significant environmental associations so far. For the comparisons of selected families with natural populations, we use these two breeding zones in BC, the eastern in pink here and the western in green. And so I'll show you the data based on those two breeding zones. So for the selected families, did have higher annual height growth for both the breeding zones. And this comes with later bud set. But in the drought treatments, those effects were greatly reduced. So drought decreased height growth and induced earlier bud set. We didn't find any strong differences in the natural versus selected families for fluorescence declines or for cold injury in either of the breeding zones, except for in the eastern zone, there's a slightly elevated cold injury in the selected families. So just to summarize some of the things I've found so far based on the common gardens, we see high variance within and low variance among populations, which indicates relatively weak local adaptation in this species and high adaptive potential. The climate relevant traits associate most strongly with longitude and elevation, reflecting some of the climatic gradients along those geographic axes. And drought resistance had the highest variance among populations, but we haven't found a strong environmental association so far. Selective breeding for faster height growth delayed bud set, but we didn't find so far that the breeding programs have had strong effects on cold or drought adaptation, but maybe some potential there for cold adaptation with further breeding. For the drought treatments, reduced overall phenotypic variance and the height growth gains in the selectively bred families. But one of our lingering questions is, are those gains, can they be recovered in a better year? And some practical implications for assisted migration of Western larch, at least in British Columbia. They're going to move into these areas where the climate is expected to be more suitable. That's depicted in the yellow and the blue here. Is that it's going to be important to consider spring frost free date, especially since larch is a deciduous species and it flushes in spring, all of its leaves. That seed source may be less important for cold hardiness compared to the other conifer species that we've studied. And then moving seed from eastern populations to milder climates could yield higher height growth gains, but this likely is going to depend heavily on drought risks in the future. So for some of the ongoing and future research that I'm planning, I'm looking at drought recovery this year, and here's a current picture of what the treatment differences look like in those beds. And then I'll be looking at trait heritability in the breeding families, genetic correlations among those traits, phenotypic plasticity among the treatments, and genotype environment associations, and looking also at what genomic prediction can tell us. And so with that, I just want to thank everyone who made this work feasible and fun, especially in the tough times during the pandemic, and all the funding sources for this work. And I probably ran over, so I'm not sure.
So we're a little behind, so thanks.
Thank you.
everyone.
Sorry about that.
Lots of questions we can ask you, we'll have to find out. All right, Emily. Right here?
Yeah.
And let's see.
The PDF solution just be able to.
Just move it over and
Yeah.
then. Come on. Okay. All right. So Emily Grau from the University of Connecticut is going to tell us about tree genes and cartography.
Can you guys hear me okay with the mask on? Okay. So I'm going to talk to you today about tree genes and cartography. Tree Genes is a web database for forest trees, and normally at this point I go into a spiel about why trees are important, but I'll skip that. So we bring together publicly available data for forest trees. On top of that, we layer curation by human or many humans. And then on top of that, we have tools for analysis for our users that they can use from start to completion right on their website. Where does this data come from? A lot of our data comes from other primary repositories like NCBI. We also bring in data from Dryad, and we have direct user submissions. So the user submissions that we're bringing in, these are things that are not being held by places like NCBI, or maybe not standardized like you might find at Dryad. So I said it's for forestry genetics. So we are holding really anything related to genomes, genes, markers, etc., but also phenotypes, environmental data, and some related data like literature or things for the community like conferences. And what I'm going to do is I'm going to walk through a little bit about how to use the website and then show some of our analysis tools that we've been working on. So the first thing you might want to do is a standard search. It's up there in the corner, might be hard to see. That's Pinus. You get results, you can click on them, but there are other ways to access the data. So we have a species overview page. This is the sequence summary by order. We have 16 different orders, 250 families, and over 2,000 species in the database. And here you can see summaries of numbers. So we have genomes, transcripts, we have genes and proteins, of course, and we have markers and phenotypes and our unit genes. You can also drill down to the individual species. And from there, you can click out and look at a species profile. We also have a species page where you can go look at genus and search. And this is what you would be brought to. So this is the species overview page for sugar pine. You can see the tabs up there that allow you to click through to the data that we hold. Sugar pine, of course, has a genome. Most of our species do not. We have, as I said, over 2,000 species, about 180 genomes, which is amazing, but most of these don't have annotations. So most of our species are not going to have a genome tab, for example. We're just going to show you what we have available. But for sugar pine, we'll look at a genome tab. You click there, right away, you can see the files that are available for download. There's something you can click out that shows you the details about this genome. And what I've highlighted with the circle is a link that you can click that brings us to one of our tools, JBRAS. Ta-da! I'm going to go back, and we're going to click on this other tool. This is our sequence similarity search. It uses Diamond to make things a little bit faster, in addition to traditional BLAST. And this is one of our analysis tools. So from our website, you can provide your sequence, what you want to search against, and all of that analysis gets done on our end and comes back to you. I'm going to use that as a jumping off point to talk about our user accounts. So I know everyone, on the internet wants you to sign up for a user account. I had to sign up for one to pay for parking at a national wildlife refuge. And it asked me for my birthday, which we do not do. Our user accounts are easy and quick. And it allows you to use our tools and come back. There's the analysis section, and you can view the analyses that you have run and get your results. I'm going to come back to our user page. This is also one of the places you can start a data submission to us. So we've worked on a pipeline to allow users to hopefully easily submit data to us. We worked really hard to make it easy, but also to make sure that we're collecting enough information so that it's useful for other people. It's called TIPS. And to that end, we have used MIAPI, the minimum information about phenotyping experiments, to standardize how our phenotypes are brought in. MIAPI utilizes ontology terms. just standard terms that can be shared across experiments so that we can connect data from different experiments. And we have similar steps for our genotype and environmental information. Once your submission is complete, we look at it and approve it, and it goes onto our data submission page. If you click on one of those, it brings you to the overview, where you have a lovely map, and there's an accession, so you can use this for your data accessibility statement in Universe. And it's a place where this data will live forever and people can access it, and hopefully it'll be useful to someone else. From this overview page, you can click through, see what's available, phenotype or genotype information about the paper, the species. And go back to the species overview. This is the population genetics tab. And this is where our tick studies are displayed by species. And you can click through here to our big analysis tool that we've been working on a lot, Cartograph Plant. So Cartograph Plant is a web-based resource that brings together genotype, phenotype, and environmental data, and allows you to do landscape and association analysis, all again from our computational resources. The data for Cartograph Plant comes from what we just talked about, but we also bring in data from BN and from TreeSnap. And we have brought in over 900 different environmental layers. So I'm going to give you a quick visual tour of using Cartograph Plant. Hopefully you can see there's a map with the dots representing our trees. Here's a slightly closer view. We have an environmental layer open, and there's a tab telling you about the layer. Here's another view. This one includes whirlwind and information box has popped up based on where it was clicked. So here I selected a tree, and from here you can view the study that it came from. It tells you what type of data is available for this study. I made a collection of trees here, and I have clicked through to the analysis section. I should note that this is still under development, but it's super duper close, and we're going to be doing a workshop about how to use Cartograph Plant in a week and a half. So from here, you can filter. You can filter by trait, by phenotype. You can filter by genetic marker. There's a few more that I'm going to skip because I'm not close to the end. And you can select what data you want from the environmental layers to be brought into your analysis. And then you can select the workflow that you want. These have been curated, pre-designed by us to fit the forestry community research needs. And it'll be selected. The available list depends on what data you have already selected, what markers are available, etc. You click submit, it runs, it comes back to you on your user account, and hopefully something good happens. Okay, thank you.
Okay, so Cartograph Plant is going live next week, is that right?
That's a loaded question. We're having a workshop to run through it. So we have been working super hard to get everything up and running.
Cool. All right.
The 24th. Thank you.
All right, Jason. Right there.
Okay, so Jason Howard is our next speaker. Take it
Thanks.
away.
Looks like I've only got 3 minutes until the next thing, so I'll try to be quick. Okay, so you already had a nice background from Jared on American chestnut breeding and restoration efforts. I'm going to focus in on one narrow aspect of that, which is our efforts to understand natural genetic diversity in the wild, in those stump sprouts that remain for chestnut, and to try to get towards a strategy for conserving that germplasm. And this is mostly work by PhD student Alex Sandercock, who couldn't be here. He's actually doing an internship this summer at a company. So our ultimate objective, as I mentioned, or as Jared talked about briefly, is we want to increase the effective population size and the adaptability of those, especially the transgenic chestnuts, the blight-resistant transgenic chestnuts. So through crosses with wild pollen. And so how do we get there? What we decided to do was to use a whole genome sequencing approach to understand neutral and adaptive diversity in that sort of remnant populations of chestnut. So we over a couple of years, we sampled 384 of these wild stump sprouts of American chestnut. A few of those were large surviving trees. We also sequenced 95 Castanea congeners, so all of the other Castanea species at about 20x. Actually, it turned out to be about 17x. And so this is just illustrating all of our sampling sites. The blue triangles are the dentata, the American chestnut, and then some of the sympatric congeners, Castanea pumila, and a couple of subspecies, alabamensis and ozarkensis, which are these three sort of recognized groups of chinkapin in North America. So why do we need whole genome sequencing to effectively sample this wild adaptive diversity? Well, my hammer happens to be genomics, but there's a more important reason, which is that these stump sprouts in the wild hardly ever produce nuts, and so we can't do conventional provenance tests in order to understand the distribution of adaptive traits across the range. And so we did all the sequencing, and the first thing that we did was to try to understand whether any of these sprouts that we sampled were hybrids. So don't worry too much about the coloring of all the dentata samples. I'll get to that next. These are the congeners, and the question is, were any of these quote unquote wild samples, are they hybrids? Because there's a long history of planting these exotic species in the US. In general, no, we did find a few sativa hybrids and a couple of Melissima hybrids, so Chinese chestnut, and a couple samples that were actually looked like they're chinkapins. A question for a later date is, what is the nature of these three chinkapin subspecies, alabamensis, which is in northern Alabama, ozarkensis, which is in Arkansas and Missouri, and pumila, which is the typical chinkapin of the Appalachians. And so it looks like ozarkensis at least has some contribution from one of the dentata populations, so there might be some introgression or something going on there. But we're going to be looking more deeply at the comparative side of this with some other analyses, not that I won't talk about today. So what did Alex find when he used admixture? He finds that we can roughly divide Castanea dentata American chestnut into three populations: a relatively homogeneous northeastern group, a central group that encompasses the sort of center of diversity of the species, so along the spine of the Appalachians, and then this distinct southwestern population in the warmest part of the range. Interestingly, if he allows for another cluster, what we get is a separation along the spine of the Appalachians, such that the east and the west, or the east and the west side of the Appalachians become sort of two separate populations. And so that may indicate the history of post-glacial migration up and down, or up either side of the Appalachians as they moved out of their glacial refuge. So like many tree species, chestnut has experienced periods of decline and rebound through the Pleistocene. So going back 2 million years, we see this decline in effective population size, and then sort of up and down as the glacial cycles sort of ebbed and flowed over the last couple million years. Since the most recent glacial maximum here, the effective population size has rebounded. Interestingly, so this is that southwestern group ends up having the largest effective population size. The central group, the northeast group is second greatest. We have this little dip in the central group, which might be just a statistical kind of artifact or something. That we don't expect to see a decline as a result of the blight-induced bottleneck, because there has been no sort of subsequent drift and evolution of the species, because there's no generational turnover. We see clinal variation in nucleotide diversity and the frequency spectrum. So the southwestern population on the bottom here, the bottom panel here, has the highest diversity in spite of being sort of the geographically most constrained and smallest population. So that's indicative of it being a glacial refuge and a melting pot of diversity over a long period of time. And by contrast, the orange line here is the northeast group, and it has the highest diversity. If we look at the frequency spectrum summarized with Tajima's D, we see that the southwest population has the most negative sort of average genome-wide Tajima's D, and the northeast has the closest to zero. And this is indicative of the history of bottlenecks, with the southwest being the most ancient population bottleneck, and the northeast being the most recent. And the reason I say that is because if we look at the distribution of the allele frequency spectrum across millions of alleles, what we find is that for the bin that encompasses low frequency alleles, the northeast population has a relative deficiency of low frequency alleles. And that's what you would expect if there was a recent sort of truncation of the gene trees as a result of a population bottleneck. So southwest, relatively stable long-term effective population size that has a wealth of both rare and common diversity. In the northeast, a recent bottleneck associated with post-glacial recolonization. One of the ways that Alex started looking at natural selection was to use this raised D statistic, which is kind of a composite of a variety of different statistical tests to look at the history of natural selection. I won't talk too much about this because I've already talked too long probably, but one of the interesting things that come out of this is that among all of the selection outliers for the three different populations, the thing that is most significant in terms of gene ontology terms is defense response to fungus. So this doesn't likely indicate that that genes related to, say, chestnut blight have been targets of selection, but there is a history of selection related to fungal pathogens across the chestnut range. And these genes might be interesting to look at in the context of blight resistance, in spite of the fact that we're not saying that they are sort of direct targets of the blight. And so one of the primary goals of this work is to sort of circumscribe adaptive units across the range and decide how to sample that diversity for the purposes of breeding. And so Alex did a redundancy analysis to try to look at the multivariate genomic space that comprises adaptation to environmental variables in this species. And so we find that the greatest contribution comes from temperature and precipitation variables in terms of selective constraint. And the second most significant is evaporative demand. So the combination of heat and moisture in the warm parts of the range appear to be an important driver of adaptation in chestnut. I don't have time, obviously, to explain how all this works, but we can talk more about it later if you like. In terms of how climate change can be expected to affect this species, now the samples that are on the ground we don't care about because they're all just stump sprouts. They're not going to adapt. They're not going to further evolve. But we do care about how we deploy this natural diversity on the landscape in the future from the crosses with the blight-resistant breeding samples. And it looks like, as you might expect, in the Northeast especially, we're going to see a significant mismatch between what is the diversity that's presently there and the environmental conditions in the future. So ultimately, our goal is to vegetatively propagate a large number of samples from across this historical range on the basis of dividing the range probably into 3 sort of management units that are defined by the neutral population structure. And then within those management units, trying to subsample them into relatively homogeneous groups with respect to their adaptive allelic content. So yeah, this is obviously work of a large number of folks that help with sampling, with the lab work, and developing that reference genome that was invaluable for doing this kind of analysis. So thanks.
Have you done demographic features reconstruction to know if the effective size has maintained a long time?
Yeah, so that's what. So I kind of glossed over that, but that's what this one would be. So this is the SMC++ analysis, looking at going backwards in time and the effective population size. And so this is so far back I wouldn't even worry too much about it. But you know, at the end of the glacial period in gray here, the population size goes up. At the start of this glacial period, it goes down, and then at the end of the most recent one, we see the population sizes going up again. Yeah. Sorry, I was going way too quickly through all this. So yeah.
How did come to the conclusion of crossing the pandemic?
This is based on simulations that Jared did to try to get the effective population size up to 500. And so maybe you want to explain that a little bit.
Yeah, just based on the amount of inbreeding that I expect from doing intercrossing after doing that outcrossing to wild trees, and then the effect population size. That I was just trying to figure out what's the minimum amount of effort we need to do to sample that wild population. And I think this adds another dimension to it, but it's still like figuring out the efficiency of like, are we capturing all the gap in space with our collection of important tests?
Yeah, seems like you have a very clear case for assisted migration or parental selection and breeding.
Yeah.
You could use those southwestern alleles.
Yeah, and I think that, and so I skipped over a lot of that, but yeah, I mean, I think that capturing, we're going to have to do some disproportionate sampling in the Southwest, because even though it's a small part of the range, geographically speaking, it's a wealth of diversity down there, and it's diversity that's going to be important, as you say, in the going into the future. It's the diversity that's related to adaptation to, you know, warm, high temperatures in the summer, and also the interaction between temperature and water, which I think is really important. Yeah.
But from an adaptation standpoint, temperature and moisture, how far geographically would you need to go right now for 20 years out? 50 years out?
How far would you have to move a sample? I don't know about the exact numbers, but well, so yeah, I mean, I don't know if I could tell you on a kilometer basis right now, but yeah.
So if you were certainly southwest, alleles would be useful in the center.
Yeah, definitely. I don't think you're moving alleles from the Southwest up into Maine or something like that, of course, but and it's complicated because the Appalachians, funny things happen in the Appalachians because you have, this high elevation, more northern-like environment. And so like, it depends on exactly, what your elevation is in addition to what your latitude is.
Okay, Jason, thanks so much.
Want me to start? Okay, so after a whole bunch of really good talks, we're now this afternoon going to move to what I would call more strategic type sessions, which is going to be really sort of the ongoing, a large part of the ongoing mission of the society is to do, be constantly sort of evaluating the health of the discipline and anticipating directions that the discipline might be moving in response to things in modern forestry. So to begin, today's session will be what we're calling topical sessions. To break out into 4 different groups and to just have a brainstorming session together about sort of the current status of the topical area and where that we think that topical area might need to evolve or advance to meet the challenges of the future. Tomorrow, we're going to be taking on what we call standing committees. Those will be the sort of the functional bodies of the society. But we thought we would begin with sort of taking a good hard look at the things that we do, the things that we've heard all morning. So we're going to break that into 4 sessions. There's going to be leaders for those topical groups. Are you putting up, Jill, there?
I am, yeah.
Where they are? Yeah. Who's doing what, where? Okay.
Yes.
And we'll get there in a minute. But I guess we're going to use what, an hour and a half, something like that, for those topical sessions. Hopefully the group leaders will assign a note taker that you will be able to come back into full group session following the breakout sessions where we'll do maybe a 5+ minute reporting from each of the 4 groups. We'll bring all the group leaders up here as we did earlier today with the panel discussions, have those 4 reports, and then we'll have something similar to the panel discussions that we had this morning, where we can just have a give and take collective brainstorming session with everybody in attendance. Does that sound like it would be fun? Let's hope that, and all of that we will capture, and this will be sort of raw material that will go into, what we hope will be a fairly comprehensive white paper for the discipline that will come out sometime on this meeting. So let's get to the business here. We didn't pre-assign people to sessions because we want you to sort of self-select, but we would like to get roughly an equal number of people to each of those. So can you just take 1 second and think about the session that you might like to attend, and then I will ask for a show of hands for each of those, and we'll just take a quick look around the room and see how balanced those are. And if they're very skewed in some way, we'll iterate it and see if we can get roughly, it doesn't have to be exactly equal, but let's make sure we don't have everybody going to one and nobody to the other 3. Does that
Sounds good?
about what we talked about?
Yeah.
You can see we're improvising here. This is not highly scripted.
So maybe we should have some guidance about what do we expect from those breakout sessions, Like, what's the goal here? You mentioned white papers, but what are they going to be about those checklists goals?
Well, I was going to leave that a little bit to the leaders of each of those sessions, that you would begin, and hopefully the leaders are prepared to do that, a little bit of an introduction to the people that have attended your session. But, as I might have said earlier on one of our phone calls here, I didn't want to put any hard expectations on what should result from these. In fact, in my lifetime career, some of the greatest moments in science were very much serendipitous. And to let, a whole bunch of smart people, sit together in a room and have at it and see what comes from it. I guess I enjoy a little bit of chaos. But I think chaos leads to good science sometimes. Because if you put the expectations on it, then you put boundaries on things. And I would rather not put boundaries on anything. I would like all kinds of crazy ideas to come to the front and we'll sift through it and see where we want to take this discipline into the future. So hopefully that, are you okay with that, Rick? I know you got busy, but yeah. That's fun. Yeah. I think the main thing is to be creative and show a vision for the future in your discussions and not be constrained by anything that we think we should be doing or what we're doing today.
I think that's what we expect from the other side, that there will be some overlap between the different ethical sessions. So if you miss something in one, you probably reach some point where you will like generic and database resources are everywhere. So I expect that you might highlight some of the items that we see in the other session. So you won't miss anything, and we will continue to share the document. So don't worry about missing something.
Yeah, really what we're trying to do here is just capture content in terms of people's view of how our discipline is performing in a subset of areas and where it might be going. So with that, I think we can move toward trying to break out into 4 different groups.
Did we want to do a quick raise of hands?
I was going to do a raise of hand and just take a look around the room.
That sounds great.
Will that work?
Yeah.
All right, so let's just start with that. How many would like to attend that first session on genomic and database resources? 1, 2, 3, 4. Yeah, we have about 60 plus people here, so maybe 15. That looks pretty good. I think that, I didn't count exactly, but that looked like roughly a quarter. So leave that for now. Let's see how about the second one, field and genetic test. 1, 2, 3, 4, 5, 6, 7, 8, maybe a little on the light end. So if we get a little bit heavy here, think about switching over to one of those, but that looks okay too. Breeding, that's going to be a big one. Yeah. 1, 2, 3, 4, 5, 6. Okay, that's probably pushing more than 15, moving a little bit toward 20. If there's anybody, we'll just leave it. If there's anybody who raised their hand for breeding and could feel like they could contribute over into the, is there anybody who feels like they might want to leave the breeding one and move over into that second one? Carol, your one? Okay, I think that just did it. I saw Brian and that Patrick back there. Those guys are like the biggest breeders in the world. I guess maybe they're a little bit tired, but like do something else. So maybe by default, we're going to be okay here. Let's look at the fourth one. How many, see a lot of interest in that. What do you think, guys? I think
we're good.
Yeah, no, this is just what we hoped for, that, this process would be somewhat organic and we didn't have to do too much shepherding. So,
yeah, hold on.
Yeah.
Each of the leads is standing up here. All right. Sure. Here you go. Sure.
That one works pretty well.
Yeah.
That's really all it was.
We do the group, what is that? Oh, people coming in tomorrow or? Oh.
All right, time to get started.
What's that? Hopefully, yeah.
Hi, everybody. Perhaps please have a seat. And I would like to invite, the lead of each of the topical session to come in front. So on my behalf, Jill Hamilton will be there. Otherwise, pick up. And Nick and Ziki. Who's going to be there? Nick or Ziki?
Here comes Nick, Zaki.
Nick.
Yeah.
Okay. Otherwise, Jill.
Jill Hamilton.
Jill Hamilton, please. Where is Jill? Okay, she's there. Good.
Okay, I'll say a few things.
Okay. I can sit in.
Okay, thanks, everybody. Yeah, we wanted to get started so we don't have to cut into what's going to happen at 5 o'clock, right? Nobody wants to miss that part of the program. Okay, we're going to begin, I think, with short reports from each of the, we probably should try to keep those to about 5 minutes at best you guys can. Maybe we'll hold questions. We'll let each of the 4 reports, and then it's just an open discussion session with the panel here. So be thinking of things that you might like to ask some of the other groups which you did not attend. I think that's probably how it will go. Okay. So who wants to start? Was it? I think Jill. We'll start from the top.
Hello, everyone. Just pulling up my notes from our session. So our group was focused on genomics and database resources and thinking about the strengths, the weaknesses, the opportunities, and the threats. We sort of did a SWOT analysis for our team up into groups and got some feedback. I try not to influence the discussion too much. And so I just want to sort of summarize a little bit of what came out of that discussion, which I think was really fruitful. Actually, the strengths were the hardest. The hardest for us. We're very critical of all the things that we need to do in the field of genomics and also in databases as well. But trying to look back longer term, we really saw that we have now the ability to look at non-model organisms, right? We're working with a lot more genomes than we had before. That is definitely a huge strength in our field. And as a result of that, we're able to do more with comparative genomics, moving in the direction of pan-genomes. We're also able to use more machine learning and AI type technologies in with these very large biological data sets that we have. So we're seeing more and more of that happen and the ability to integrate data as a result of that. And of course, there's a lot of focus on just the technologies themselves. That's a strength. We have long-read sequencing, we have IC, we have optical mapping, we have all these tools now available to us to, attack more complex and larger genomes. So that was our strengths. And then on the weaknesses side, there's still really large complex genomes. They're very expensive and they're very difficult, and we don't have enough. So if you look to the conifer community, for example, we still have a relatively low sampling rate of the diversity of species that are out there. And for certain groups, the same side of the strength is a weakness. Comparative genomics is still challenging, especially for some of our larger genomes. And we also have just challenges in terms of the analysis overall. Reproducible workflows is a really common theme of being a challenge in our field. Also, where we can even document and find those reproducible workflows. Funding came up a lot as a huge challenge, especially on the database side in terms of thinking about reliable and stable funding related to maintenance information. So there's lots of opportunities out there to develop new tools, new functionality, but there's very little out there to think about how do we maintain the integrity of data that we have. In terms of our opportunities, there's a lot that's going to happen in the future for data integration, and that means not just the omics, but also phenotype and also environment, and that's an area that a lot of groups are working in right now, and I think that's where the future is particularly bright. There's also just the fact that we're getting better at standards. There's a lot of new standards in metadata that's becoming more important in the field, things like FAIR and trust standards. And so there's a lot of opportunities to integrate that and facilitate data transfer as a result. In terms of threats or challenges that we wanted to call, the fact that there's so much data is a challenge, right? There's so much data, the technologies are changing very rapidly, the formats of that data are changing very rapidly, it changes almost every other week. And so as a result of that, it's incredibly challenging. There's also still a lack of kind of reliable standards in some types of this data. So we're just collecting this information, but not necessarily integrating it in any reliable way. And I think a lot of what we talked about also was a need to really have a connection between journal to long-term identifiers of data directly to robust pipelines and analytics. And so we still really don't have that full ecosystem, partially because of the large amount of data and how things are changing. But these are going to be continued threats and challenges moving forward. I think that summarizes everything we talked about.
Thank you, Neil. Yeah, we'll just keep moving and have a discussion on everything else. So now it's either Nick or second vote. Yep. No. Victor, you're next.
No,
Well,
you are
we'll
the
just
order.
order there. I'll pull up my technology here. So we were given the task of looking at genetic resources, genetic tests, etc. Is it the zone?
Yeah.
And we use the same coordinated system, the SWOT analysis. There was a tremendous amount of information provided, an excellent group of folks contributing. We captured a lot of information, and I'll try to keep it very short, try to identify, prioritize the information. To begin with, we had strength. The strengths, of course, are that we in the industry and our institutions have a tremendous resource of progeny, genetic tests, clone trials. And with trees, of course, the information are the plants last for a long time, so they're out there for long periods, unlike your annual crops and whatnot. We make big investments to begin with in maintenance, but the population genetics are there for a long time. We have several different kinds of trials, of course, the progeny genetic test, the Providence garden trial, the common garden type trials, the adaptational trials, which are becoming more prominent. And of course, association, QTL, and sentinel type populations, which are more recent, but also of great potential value. That I would consider to be our strengths. Weaknesses, of course, and this is where we came to the realization we have, of course, issues of institutional memory loss, people retiring, forgetting where tests are, lack of the ability to maintain tests for very long periods. So the cost and the input management and cost are great and not often supported by institutions. More importantly, there's no centralized, we've concluded there's no centralized national or international databases similar to things like you've got with tree genes, for instance. And we see this as, many of us have talked about this in the past, but it's, you not only potentially lose plant materials through the loss of support for supporting the test, but you lose the data because it's sitting in somebody's file cabinets at home. There's a lack of that. We notice that, looking at the future, there's a real lack of adaptational trials outside of the natural range of the species to deal with, future potential threats like climate change. So we spent, I think, most of our time talking about opportunities. So, and we concluded relatively a number of things, but I'll just concentrate on what I think are the most important. Zeki can correct me if I'm wrong. We felt as though it's really important to have a national database facilitated by individual organizations. In the U.S., that would be the the Forest Service, and presumably there are commensurate organizations in Canada and Mexico who could provide the same service. We felt that it was really important maybe to start developing things like experimental forests where or experimental sites where people established long-term trials that were under careful management. Again, probably in the U.S., supported by somebody like the U.S. Forest Service, where you would establish, association trials, programs trials, adaptational trials, and they would have somebody responsible for looking after them for long periods of time. By that, we mean, 25 years or longer, potentially. And of course, threats are those that you all know about already: climate change, insect and disease, fires and floods, all the issues that suggest that continued investment in these genetic resource populations are critical, are more critical than ever. So I'll support Brad's statement there. Zeki, you want to add anything?
No, thank you. You summarized well, but if you'd like to know the details, we put them all on these white, big papers in the bed somewhere. We could share with them. I think this national database is strongly supported by our group that could be accessible just like NCBI, people uploading and getting free data from there. Keep track of all these past, the data collection experiments, collaborate with the future experiments. It was really good group, especially experienced people there. We got most out of them.
So we talked about breeding. Again, we followed with the basic definition. What is it to be a breeder, tree breeder? Strengths and advantages, the challenges, and maybe some possible solutions. First of all, we felt that we talked about being a breeder. It's really getting the best of the two worlds from animal breeding results, that's the methods, animal mixed models methods and pedigrees. And from plant breeding, we get experimental designs. And then we use genomic resources to make breeding more efficient. So those are not very well known in the community, that to be a breeder, that you have a lot of advantages to be a breeder. Another maybe strength, we talked about the genomic age. We are now experiencing genomic age since 2008. It's impacting biology, medicine, and agriculture for sure. Plant and animal breeding. We talked about how to access or develop and access to genomic resources. But one of the challenges we talked about in length was we do not have enough trained workforce coming from the academia to fill a lot of positions. And that was the biggest challenge we talked about. And a lot of, our graduates, they take jobs with seed companies. My student got a job with a cattle breeding company. So that's a big challenge. Except maybe one or two universities, a lot of, programs, either they close down, they do not replace scientists at those universities in Koch genetics and gene. So it's a challenge. Maybe, there's, society can do something about it. A lot of those programs, they need to be accredited by SNEF, but ASF doesn't require any training in genetics right now. Then we approach them and then maybe, convince them to support such training at universities. Funding definitely is one of the challenges we talked about. There is such a lot of competition to get funding and support for breeding. And breeding is such an interesting profession. You really need to have a lot of skills. They understand some genomics and grafting and who knows, root cuttings. And it needs to be a team work for any breeding program, for like ecological restoration. A breeder cannot do everything. It needs to be a teamwork. Without a teamwork, just doesn't make progress. You can start. We talked about managing the breeding research and genomic resources. We talked about CRISPR, its role, whether it has a role or not. There were different opinions about biotechnology and CRISPR. But one thing was clear to the group was that for breeding to have really impact, we need genomic resources, genotyping tools that are inexpensive, affordable, easy to use. And that was my opinion that, what we really need in terms of genomic resource for breeding, we need to have good reference genomes for species. Because reference genome is the basis for everything, from market discovery, QTR mapping, annotation, imputation of the missing genotypes. You have to have a good reference genome to do that work, to make the breeding more effective. I'm sure I am missing some other, concerns, maybe solutions we discuss, but maybe they will come up during the discussion.
Okay, and so we were part of the conservation and restoration group. And I just wanted to start off by saying that this group was really well represented by Canadian researchers and managers and US researchers and managers, but there was no Mexican counterparts in our group. And so it was immediately noted that that was a substantial gap and a potential opportunity. When we think about the strengths that we have in the conservation and restoration of genetics community, we think our knowledge is highly applicable to resource managers. This may not always be the case. Managers may have different opinions on this, but so it's interesting to think about where that translation is from. We think our research is highly applicable. Is it being applied? We think we have a strength in collaborative research. Genetics is used in many other fields. We collaborate with silviculture, ecologists, plant pathologists, and more. Another strength is we have a long history of knowledge and research and provenance trials and collections of seed resources. And just thinking about Richard's talk today earlier of the various resources that are available, I mean, we do have a lot of great resources available for the community. I think some of the weaknesses and challenges that we identified were in forestry, we think in the long term, and often funding graduate student projects, master's, PhDs, they require short-term deliverables. So how do we translate that short-term to long-term for forestry conservation? There's also shrinking number of educational programs at universities and loss of students to other genetic specialties. We are challenged to translate complex ideas to non-geneticists, and we need better ways to communicate to broader, wider audiences to ensure that genetic knowledge is translated to the range of stakeholders that we engage with. So we're going to get ideas in this group. Some of the opportunities we identified, we have opportunities to create reciprocal student transplants across 3 countries. We like the idea of thinking about this as a reciprocal transplant. We thought about cross-border collaboration to address climate change challenges. We have more opportunity here to think about this in the context of climate adaptation. We have opportunities to work together to standardize collections across ranges, across countries, to standardize phenotyping and standardize genomic assessment of collections and use of genomic tools. And thinking about kind of those threats. If we don't act, what are the threats to our community? There'll be a lack of sufficient training for the next generation of forest geneticists. A potential downward spiral and the loss of genetic diversity and a threat to the effective knowledge translation will lead to poor decision-making by policymakers. And there's more, but you can open the discussion.
Okay, does that conclude the 4? Okay.
Okay, so, sorry. Now is our opportunity to just have some open discussion, frankly. One comment I would make is listening to these presentations a minute ago, I already thought of probably 3 or 4 more standing committees that this society might need or could serve our discipline well from things that other than the standing committees that we'll talk about tomorrow afternoon. So that's a topic for future meetings. So I will really just identify people who would like to either make comments or ask questions of any of the leaders of these breakout groups. Can we start? Does anybody have something? Sally, thank you.
I have a comment.
Okay, well, I'll go to Sally and then back to you, Natalie. How about that?
There's some linkages across these buckets that we need to think about. One that comes to mind is, with genomic approaches, we always have to have validation populations. And so it's those field tests, the resources that provide us with those independent materials for validation. And it's the genomic and database resources, where those genomic tools, markers, referencing, whatever tool is. So I think it would be helpful to think about maybe those cross-cutting things. I'm sure people can think of things more than that these are not independent.
Yeah, and by the way, it's 4 buckets that we came up with off the top of our head just to begin for this session, but of course there would be many others that would apply to the discipline. Natalie, were you wanting to make a comment or?
I fully agree with Sally, what you said. I think it's gonna be over the conversation over the coming years that we perhaps come to a type of agreement or something, how we can do it. But I will have a question for Peter, because you mentioned that reference genome are very important, but on my side, perhaps I don't know, for you in the US, is this something funding, but on my side, looking at it, doing new provenance trials or common garden experiment, there's no money to do that. So how can you justify, getting money for genomics? It's easier than getting money to do and develop, for example, precision phenotyping and develop new trials. So I think that this is something that is really, I see we are really poor. I don't know how many field trials have been put into the ground recently the last 20 years, but on my side, from CFS, there are very few of them. They are rare. So I think that one of the challenges going to be with what we see is when the trees are growing where they are major, they are less perhaps affected than the younger trees. So, and there is a whole gap there. So I just want to.
So then we made a good point that, without really having a population to work on, like a breeding population, it could be for food production, could be ecological restoration or disease resistance. Genomics not gonna be useful. You have to have a well-defined managed breeding population in place, trials. That's really the basis for everything. So I completely agree.
Okay. But what about
But,
other rest of the species? We are talking about conifers, but for other species than the commercial species, how would you?
Right. So, genomics is another topic. If we have resources to invest in genomics, resources for breeding, that's what, we talked about. Maybe, developing a reference genome for those species would be a really good way to start with, to develop other resources, genotyping platforms, for example. QTL discovery, annotation, they all follow. But I agree. If you are working on some niche species, minor species, ecological restoration, put together a well-defined, small management population to make sure you turn the breeding cycles faster. That's the key. And not taking on very large populations and you don't make progress.
Yeah, I would just make a comment that I made, I think, in your group, Vikrik, is that I think all of the groups are sort of pointing to, lack of support, funding, resources, the things that we need we know how to do it. We're just not always enabled to do it because of limitations. And in my experience in this discipline, we tend to do it, individually, one at a time, and really, I think what this discipline needs to be thinking about is, going to the moon kind of applications, where, instead of, one genetic sequence, one common garden, as we come together here, think about, big things, because these are the things actually that get attracted to funding agencies, because, it's the kind of things that politicians can get behind, because they can get credit for very big ideas. And I think where I was accused of it very early in my career, you're thinking too small. And I think as a discipline that it's a little bit systemic to us. So I would just throw that out in your own thinking here is, let's not put any boundaries or restrictions on us, and let's think big like other disciplines do.
I have a question and a comment. The biggest silo I've ever seen at the university was between agriculture and forestry, and how it would be kind of cool because they're doing everything we've done in the forest side of forestry because it's not an annual crop. So they're like not listening. But if you can get them, some folks who really understand agricultural systems, I think we could learn a lot from them. Some integration, like we're not going to win them over, but we can learn from them. And there's just no bigger silo I saw at the University of Minnesota than there was in forestry and agriculture. I was struggling with things that they were like, had no problem. So how can we learn from these annual crop people and to help make our discipline a little bit? I mean, we have different outcomes involved with a lot of different technology, the database, database mapping sessions, some real basic stuff is similar. So it'd be really cool to have some agricultural people, gene conservation. You know, think of CIMMIT and some of those other international groups that hold gene conservation. What can we learn? And is that a threat to us, or can we make that something useful to this society? Maybe bring some ag people in to kind of, hey, observe, what should we be doing?
Yeah, no, what I'm hearing from you is, could be very much a thematic topic for a future meeting where you bring in those people and brainstorm collectively with us, you know, not only to be advised, but as you say, to talk about working together in ways that maybe they've been a little bit more successful than we have been. Can we keep it going? Carol?
A couple of you brought up education as being a problem and a shortage of workers. I'm assuming you mean at all levels, undergraduate, master's, PhD. I'm wondering, I don't even know if it still exists, but at some point we had one of these USDA National Need Graduate Fellowship programs within our department. And one of those things be written by a big group where multiple people at different institutions could get funding to train one more PhD student or one more master's, and I don't know if it can contribute to undergraduate, I teach undergraduate tree improvement class. I'm just wondering if that's something that if we need a group on education.
Well, Carol, you're thinking big. I like it. Yeah, instead of you submitting one application, it's. Yeah.
I think that's a really great idea. Here's an example. I had a student, a master's student that I taught forest genetics to this last semester. Okay, I'm a terrible teacher. But there are lots of good teachers in this group. We had a curriculum that we could offer. Then I think there would be a lot of value in that, because maybe at Ohio State and the University of Minnesota, there are 2 graduate students that are interested in forest genetics. Well, they're never going to get anybody to teach in forest genetics. But if we can figure out how to offer them a curriculum, then we might get more people interested in forest
So one is an undergraduate course, and usually about 1 per year express an interest in continuing in forest genetics. And one thing that's lacking is I use Dave's book. I don't require this why I can go with the next in the course.
Yeah, it's pretty good book.
Blame the publisher. Yeah. Andy.
Well, I mean, along those lines,
These are great ideas.
I mean, it would be developing a curriculum is a great idea, but if you've got 2 students at, Ohio State or 2 students here, where there isn't maybe, a particular professor that can lead that, it's difficult to make that work. Right? I mean, but I mean, we've just gone through 2 years of virtual meetings, virtual classes, all sorts of things. I mean, why not try to develop a class that would be somehow accredited, that graduate students from across the country could take, they could get credit at their home institution where they could, of course, genetics class.
That's where I was going with this.
Yeah, are we not having a session tomorrow on graduate education? Who's leading that? You, Jill? No. Pickard, yeah. So boy, yeah, these guys have got to jump on it. Yeah, there's, North America forest genetics, not NC State forest genetics, maybe in our future.
Those are great ideas.
And populated with scientists now out of that forest in the landscape field. Yes, and the next course was great, international, and here we got together and multiple leaders out of it. So as an idea,
Yeah, with the individual universities would have to do is get a course number listed so that, because that's going to differ from institution to institution. In the European system, you just attend a course and it doesn't really, it doesn't have to be listed always, right? Whereas in the North American system, in order to graduate, you have to have, credits and so forth.
And offer it in Spanish. We need some offerings in Spanish as well.
Yeah.
We made some of the nursery manuals that Bringer produces, we've got some Spanish version of the tropical nursery manual for folks in Central America and other places. So.
Yeah, the textbook that I wrote with Tim White and Tom Adams was supposed to have been translated to Spanish. Did it ever happen? Did anybody ever see it? It got translated to Greek. We were told that you wrote it in Greek, but yeah, that I don't know why that hasn't happened. Zeki.
Make a comment. About those, lack of faculty in forest genetics schools. We had some experience in Europe and in Turkey that, you could have agreement between universities and students, go to those agreed universities, take course load and then come back to your department. Sometimes dual degrees or sometimes, just course credit assignment. I think there could be easy solution to that. Just, NC State and let's say Ohio State could agree on graduate program that courses not offered in Ohio, they could go take in North Carolina State 1 semester or semester come back. This has been practiced.
Yeah.
I would like to hear our Mexican colleagues about what are the main challenge, because I think that sometimes we, on my side, we got, if money is not the problem, it's really to get the forest manager that believe in what we are doing, because we have everything in hand to show what we can do, but it's too long, so they are not interested in that. So on your side in Mexico, what do you need to, what are your main challenges there?
I think the two main challenges, the always challenge of lack of funding. The second, question regarding security for I would say that it's 2 points. And also regarding recently I participated in a course for people from Latin America organized by the INC, Agencia Española de Desarrollo, USAID for Spanish version,
okay.
So they would everything in Spanish.
Okay.
And it was targeting people who is working. So we would ask to report our voice in our own presentation. So the students would take it at any hour that they could, and then they were doing synchronization by Zoom or a platform similar to Zoom. So it was a kind of hybrid format that we could not imagine 2 weeks ago. But we learned how to do this platform and this pandemic and I think was successful. So I think it could be possible, something like we said in the Spanish version, using this hybrid system with new resources that we have.
We still have some time. Any other thoughts that people would like to, Andy?
Well, just following that idea, I worked with the Forest Service International Programs earlier this year, and we developed a course for Central American countries, and it was on seed bank management and climate change. So it was a similar thing where there was 4 pre-recorded presentations. International programs had contractors where we recorded it, our slides, they were translated, both the audio and the slides. The students that registered for it watched the session at their, on their own schedule, and then we had a 1-hour Zoom meeting, and they got a certificate at the end. So I mean, something like that could very easily be developed. I mean, we can talk, and I have a contact in the international program, broke down, you know, there's a desire, there's an audience. I think that certainly be doing it.
I
Go ahead,
have a
Natalie, yeah.
question for the audience and for my colleagues here. Why it's so difficult to recruit people in forestry, do you think? Like even as a bachelor degree, what we see is a decline in the population of students. They are less interested by forestry. And I think one of my explanation is that forestry is often seen as a kind of a traditional thing and not very technology driven. But do you think it's the only reason for that? Or because I've seen when I talk to my colleagues from other departments, all the time when they explain what we are doing in forestry, they have a very, I would say, it's a kind of characters, it's very far away from what we are doing right now. So the students are less attractive because they don't know that we do remote sensing and genomics and everything. So do you think that there are also other reasons than that?
I can respond from my own experience. At UC Davis, clearly, you know, the students would visit my office. I think many probably had the same experience. You know, and they go around, they talk to a bunch of different faculty, and they come by, and I'm honest with them, you know, it's hard. It takes a long time. Genetic resources have been limited. And after they've listened to me say that for about 5 minutes, they're right back down to going to work on the Arabidopsis so that they can get a degree, a publication, and a job. You know, it's not an easy path. I think to get a graduate degree in forest genetics and then think about permanent employment and, you know, feeding your children. And I think a lot of people young people would love to work on trees. They love them. But practicality says, maybe it's not the best bet for me. So I think building things up as we are trying to do here in many ways is going to assist that. And maybe I have your own experiences, Sally and Janice.
So we've never had so many students at the undergraduate level. We diversified our course range, our program, and it's so traditional forest management. We still have good numbers, but it's not the bread and butter program that it once was. But we get students coming into our natural resources conservation program, our urban forestry program. We have a new program that's the bioproducts and bioeconomy. And students come in with these different paths. And I will say that last program is food products processing, and they graduate with the second highest average salaries in our whole university. But that's a very technical program that continues at that undergrad level. But that brings these students in, and many of them end up in more forestry type jobs. But they come in because they love forests, they love the outdoors. They don't know who they would ski or not. But anyway, so I think that we have to, I think in some ways the word forestry is a problem. Because it's more about forests and natural ecosystems. And many of the questions we're asking, I mean, there is traditional forestry and there's tree breeding, but there's a whole range beyond that. And I think we need to be careful about the language that we use when we're reviewing students. And then at the graduate level, we have a shortage of applicants, although our cost is a challenge. But I will say, if you're saying, it's hard, but there have never been so many jobs. How many jobs have been advertised in the last 2 years? Postdocs and permanent positions, of course. How many are going on? How many of those postdocs haven't been filled that I see advertised? Repeatedly, there is demand. And so, maybe it's the way we're marketing it. Because I think that, of course, I think it's great.
Jenny, and then Swelly.
Just following up on what Sally is saying, I'm not in the faculty of forestry. I'm in the faculty of science.
and maybe something of a disconnect from a lot of the existing services of forests beyond recreation and aesthetics. And so that disconnect starts early with high school counselors who would probably not put forestry in the top 100 occupations that they would give to virtually any student. We see continuing encouragement towards medical sciences. And so that urbanization of our populations within North America, I think, Sal is exactly right that we need to, well, I hate the word rebrand, but let's just use that, rebrand what it is that forest biology, forest science, all those sides of forestry are. Certainly in our university, when people are interested in forests and they're coming from an urban environment, they're almost exclusively going to the college programs because that's where they think they can make a difference. So we do have to think that our target populations are teaching, they are way more diverse than they ever have been, and we need to reach them and reach us.
And I was gonna say that I think there are maybe not used opportunity that I have my graduate degree in forestry department and never was bought into forestry from there because our walls are hard to white. That's also a problem with forestry is that we have this many problems that get in our head of what it was. And not when the diversity kills the diversity of the word. Which is wrong. It's wrong that there's not diversity, but we need it only in the forestry.
I agree with everything that Sally and Anna said, and being in an evolutionary biology department and also not, I think, seeing the same problems. I get tons of applicants of students, we have more students than we can take. Because they're like, trees are cool, so is genomics and bioinformatics, and I would bring all of that together. And so there's just a ton of excitement about that, and the more they learn they can bring this together with a, with a job that will pay well in the future, whether they end up still studying trees or another organism in the future. So I think it is a little bit of what people have been saying, a little bit of, what is associated traditionally with the word forestry versus I'm studying trees and forests without the forestry part. Seems to make a little bit of a difference sometimes too. So, yeah.
And just as a follow-up, SAF, Society for Forestry, is just having the same conversation. They also, it's historically been very white male dominated, intimidating for women, intimidating for people of color. We're increasingly urbanized society. People are afraid of the rural sciences, afraid of working in rural areas, afraid of going to some places. And so we fall under that. And that's why because you're not forestry, it's more attractive, but this is a society driven forestry issue as well. And so we should probably connect with them and say, I think you're trying to integrate more urban community forestry. Purdue is hiring a new urban forestry faculty, just more urban forestry, finding those linkages with genetics and urban and these equal justice initiatives are those little linkages you can make and can help recruit. So.
I'd like to make a few comments too. As a forest geneticist, we should question why we're failing to effectively student or not enough students there. I think one reason is that we should learn something from physiologists, ecologists. If I look at both of these fields, they have, a well-known federation or societies around the world. They attract large group of people. So their, messages go further than their, the local community. As far as I know, getting master and PhD in Oregon State, the only the association is the Western Forest Genetic Association, and then there are, the regional organizations, but never get together. Maybe this new society that you are just about initiating, including a European or Asian federation, you would have a strong voice, that will help funding. I'm sure, forest genetics is smarter than the physiologists, ecologists, I mean, not, looking down anyone, okay? Because I apologize if I do. We should find a way, how to get the funding and attract the students. I believe, I think Dave got the, problem area and he initiated. I hope that the young generation will continue to expand this organization.
Okay, I'm going to actually adjourn because I think we're ready to be hosted for our reception. I'm seeing creativity, that's really what I had hoped for some of these sessions, not just the standard reporting of our scientific accomplishments, but thinking hard, futuring, trying to be creative about trying to advance our discipline. So let's enjoy a nice eat while we have reception and dinner. We're going to come back for like an hour or so of short talks before the sun goes down.
Yeah.
So thank you, everybody.
Yeah, thank you.
Oh, yeah, it's on the website, but we haven't done it.
Pictures. You were saying.
Yeah. You want to hold take a picture of Adam's students first? If I give you a sure. Okay.
Excellent panel. Yeah, they definitely. I just I got to do it. My son, his son are friends, and so my son did Okay, take care. No, no problem.
So now I'm not sure what that. Yeah. Other than being in North Dakota. Oh, yeah. There's a one, right?
Yeah, I have that.
Thank you. We'll see you again tomorrow. Okay. Well, good job, Dave. Well, I don't know where I left my backpack. Oh, that's important now.
to meet you. Okay. Let's see. Yeah.
Okay. I was keeping up with the thing. Okay, good. This is still working. NEFGS. Evening. Yep, Peter's is here. Thank you. Everything will go well.
Or not?
No, we're not saying that. We're not doing that.
Dan's talk. You're gonna play it? It's a video.
We can do it. I think it's working.
Okay.
It takes a while to load.
Yeah. So hers is up first. So
Yeah.
we want to set that one up.
Oh.
I think.
Oh, what is this?
Let me get the schedule.
Yeah. Hers is the first, yeah.
Yeah. Takes a minute.
I was just thinking the.
I know, I was like running over. I'm like, oh, they locked the door. I'm like, okay, somebody open it.
I was like mildly stressing. I was like, dude.
No, thank you. Yeah, it wasn't. I was like, who did that? I think it was the bar, the cocktail people probably when they left. I was like, no, it's supposed to be locked. I didn't lock it. I only lock it when I leave at night, so I was like, lock it after the session.
Yeah.
These are big. They're always big on the video.
Yeah. We've been recording the entire time, which I'm proud of.
Oh,
I thought
you did a good job. Thank you.
I thought I think in the middle it probably did, but with
like the panels and stuff.
Yeah.
That's fine. Yeah, we just need to talk. I
Wait.
figure like.
Oh. I can't hear her. Oh no. Okay.
We can play checks at the end if you want.
Yeah.
But we also have time tomorrow, so we can always just do that. So don't worry about it.
How do we pause this? Oh. Okay.
Is our sound working?
I have to figure that out right now.
But you said it worked right earlier.
Yeah.
It's so finicky.
I think it's also because in the middle there was oh, okay. It's this one. Okay. All
Yeah. And that's what you did, and it worked.
good. Yeah.
All right.
Let me see again. Quickly open this again. Yeah, I can hear her.
Okay. Can you get it louder?
No.
Is it coming out of here?
I think it's like it's because everybody's talking that you can't hear her. But,
It should come out here. Try it again. She's not moving her mouth, so I don't think it's funny.
oh.
That's the last one. Yeah?
And it kind of helps when people don't talk. So.
Totally. I was wondering if we put the microphone over there.
Oh, yeah.
Or if it'll just get feedback, which is the other possibility.
We can try. I think it'll work when yeah. Oh, I don't know what I did. It's there's a slight problem. Like what I've noticed is that she talks, and then once the slide's over, it gets over, but then you have to actually go to the next slide.
Oh,
Like,
it doesn't auto advance.
Yeah.
Okay. Okay. So is it all the way at the beginning?
Yeah. No. Now you have to like X out and then click
Okay.
the file again.
I think if you're okay with it, you can just start it for this one.
Okay.
And then I can click next.
Yeah, I can start it and then yeah.
And then just sort of introduce her as a recorded talk. And then I think, did you find everybody else in there okay? So then I saw Megan's talk, Ami's talk.
Oh, I don't have Ami stuff. Oh.
I know that he loaded it, or said he loaded it.
Oh boy.
I hope he knows he loaded it to the right place.
Yeah, it's good.
Okay.
And then Kathy put up a PDF instead of a
that's okay.
link, though, so I don't know.
Do we have sorry,
Pullingham?
Yeah. I don't see. Do I see Cunningham? No, I don't. Okay. Oh, I don't know what happened.
We don't have her. And we have Laura after that, Corona, then a Greedy, and then Haley.
Wait, I just saw Cunningham.
Could she be in the base folder?
Uh-oh. No, I don't see that.
Okay, you didn't see it at all?
I mean, I saw it like a second ago, and then I can't see
You
it.
see it in the trash? Like, did I get?
No.
We can save her to the end if we need to, and then just have her come up and put it up.
Okay, I could like walk up to her now and ask her to be uploaded to the. Yeah, I will go up to it.
Yeah, she's got time. She's
Okay.
fourth, and we can always.
Yeah, she can
skip her.
upload it again, best, but if not, yeah, we could throw it up here. if we need to. Are you feeling good? This is low-key. It's low-key. It's fine. It's fine. I won't say anything too much. I'm not worried at all. You might actually be. You're like, baby. You don't stop crying. This is like, who does this to people, right?
We started 7:10. Right. Perfect. All right, we started 3 minutes ago, so please find your seats. We'll give them a minute.
Yeah.
We'll give them a minute.
I kind of sent her back to get the presentation.
Mhm. Wow.
Let me just see if I can.
Everyone's sent just downloads so they can pull those up.
Yeah.
Okay. And you'll start.
Yeah.
Okay. We might have to holler at people to go take their seats. Because there's some people outside. Hello, we're gonna start. So maybe have a seat. Sit down. That's how I talk to my puppy. I only have a puppy. I'm out of practice. You bop on the nose. All right, thank you, everyone. So I'm Rhiannon, and I'm going to moderate this evening's talks. And we're going to start with Anne Williard, who couldn't be with us right this minute, but she has a recorded talk that we're going to try to play. Hopefully that works around you.
We'll start by doing so I'll be providing examples of recent research from my lab and others to help find new questions. One, especially involving different kind, another treats them together. So these biologies are 130 individuals represent different taxa and 700 individuals of left and details, but nuclear and constitutive. I just wanted to tell you that this distinction informs how we should be recognizing that. First of all, every time is resulting in the other red taxa and these in the army, which are the California. I'll come back to kind of step. The other is that the Ponderosa Pines West, in a strict sense, are not sister to the ones east. So Pinus particular and Pinus scapula were originally published in the USA as varieties of Ponderosa pine and the other thing I'll tell you is that we have limited within population sampling, but we'll give an example of let's talk to individuals, but an isolated standing of southern Nevada and they tell them different clades. So this is true for the easier to see the resolution of judgment time is with the codifying grade line. Also, the separation within what is traditional Ponderosa pine result back to this node B. So the Scotland bracket resolve with all of these Mexican, mostly Mexican So most of the conflict are congruent, there's a few examples like plants, Arizona. So this is where the pines east of the USA Great Basin are resolved in both nuclear and plastic. So by picture is in blue here. Those plants are found all the way up into Wyoming and all the way down to central Mexico. And most of those fragments are one limestone clay. So some first, it's mostly to the east of there, and it's not until the south boundary to the mitochondrial plate, stop that on the right, slightly different sample. Here, the is squares and stuff on the plus line, and then the colors are mitochondrial haplotypes. So most of them has to be yellow, and that's the ancestral haplotype that's going to be distributed taxonomically and geographically and central. The stop line plus line said converted to 6, and that's also been recorded up here in some samples. The other thing I'll tell you is that the extinction unknown, this is an example in Mexico, where population was assigned to particular based on mythology and on nuclear and plastic resolution is carrying the H6 habitat. So, my kind of mixing every time is amazing. So the big samples here are the ultra mix every samples. The colors for my kind of habitats. The H1 is purple, H1 is green. So notice that the Jeffrey pine samples are all of them by habitat, but either the nearby H1s or H5s. So what is trying to do is clearly different. So addition to nuclear and plastic data, the literature search showed that most all of those work. So first of all of them comes with convention setting. So different kinds of cones are not heavy and they don't have the outward ripples. They have different seed predators, so you think that we would hypothesis for why so they all have good cones. The seed movement, the seed length ratio is all less than 2. So heavier for that shorter seed wing than Jeffrey pine for means that Jeffrey pine is completely reliant on rodents or nut fibers for this work. In controversial time, we can still disperse the seed very effectively, and rodents and birds are for pollution distance dispersion. Also, every time you know, so where that energy probably helps attract animals as well as reduce effective wind dispersal. And of course, every time has saxony as a component. So that makes the Jeffrey samples carrying mitochondrial types very interesting question. So geographically, we'll turn to evidence. I'll just lay out the other questions for you. So first of all, the Pines Arizona got Pines Eagle 99. With one in the USA and Southeast Arizona. However, those names have been assigned to populations down in Mexico, and neither of these taxa are cohesive. And we also think there's at least one new taxa in Southeast Arizona or out in the population. So the contiguous undiversified kind of forest area in California, where climate cascades is here have some mixing of the coastal Megalodon, mitochondrial haplotypes, and inland continental haplotypes. There's no current sympathy, but there are 2 areas that are of interest. So in such research that you would sign that with. The interesting part of that story is that what's happening is different from the rest of the record, and they're actually closely related to the Scotland larvae, habitats that we found out here in the Northeast, and to some habitats this don't have in Mexico. There's a region of contacts and described by Whitney, and that. We also found projects on Idaho. And Idaho, interestingly enough, there are nuclear and mitochondrial sharing, but the class of haplotypes with their. And then, of course, there's time. So I just want to put out conclusion and scientific method. And that example of which plays that are actually sharing ancestry, political borders don't matter, and settings that are not. And how urban outreach habits and retention of the women. Example of that logical time, and obviously, and then people will design experiments expecting cells of trees that might have ancestry. So that is important measurement on the same plant sample, because some of the population will be clarified. So thank you very much.
So, ready? A little after dinner confusion here on my part. So don't eat a bunch of mashed potatoes and then have to come and be awake and talk to people, I guess is the moral of the story. So our next speaker is Megan, and Megan, yeah, okay. Are you gonna come up here? And she's gonna talk to us about genetic and environmental drivers of red oak phenology. Thinking about things. Just letting you guys. Okay. Do you have all the images? I have all, like, it's a 3-minute talk, but my gosh. Is it high def? Apparently.
One extremely detailed slide.
Super detailed. Just imagine it in your mind's eye. I feel like it may have stalled. Is it going? Yay! It's that first slide. Great. Okay. So this is a short talk. It's just 3 minutes. So I figured I'd give you a quick overview of sort of what I'm doing for my postdoc fellowship. I'm 2 years into a 3-year NSF PRFB in PGRB Plant Genome Research Program. And I'm studying sort of the genomic and environmental drivers of phenology in red oak, or Quercus rubras, northern red oak. And so the background here is a drone imagery of the Harvard forest. So you have a swamp over there and sort of not swamp over here, and you have all these red oaks in this image. And what sort of prompted this research was many, many years ago in my lab, someone had done all these drone measurements and looking at phenology and found that there was this huge diversity of phenological timing in red oak in this area, this one little area, that the red oaks on this side of the plot would leaf out a month later than the red oaks on this side of the plot. And I was like, oh, that's super interesting. And he found that environmental, microenvironmental parameters only explain about 15% of that variation. I was like, well, is it genetic? And he was like, I don't know, I don't do genetics. So that sort of prompted this whole postdoc that I ended up doing many, many years later. And so phenology is super important. It is a huge predictor of global climate change.
Spring now occurs 2 weeks earlier than it did pre-industrial times. That means that not only is carbon being taken up earlier, but the Earth's planet albedo is changing and water fluxes are changing, which has a huge feedback into the global climate system. And so modelers, ecosystem demography modelers care a lot about getting phenology timing right, but they completely ignore the fact that phenology has a huge heritable component to it and that it's strongly locally adapted. So what I'm super interested in doing for my postdoc is sort of parsing apart that variation, modeling out sort of using both genetic and environmental variation to understand what species range shifts might look like if you consider both environmental and genetic variation, and then I'm going to validate that. So again, I'm 2 years in, so I've already completed this, but I have a growth chamber study where it's really hard to set up a common garden of red oak in a 2-year postdoc or 3-year postdoc. So I ended up taking leaf branch cuttings post bud set and put them out into different environmental manipulations in our growth chamber and was able to force bud break. And so I observed bud break in 8 different genotypes sourced from the Arnold Arboretum, anywhere from Alabama all the way up into New Hampshire. So their provenance is from across their entire range. So I'm looking at both genetic and environment across all these different environments. I can come up with the reaction norms. I can then put these reaction norms into this phenofit model. It's a process-based model created by Isabelle Schween in Montpellier, France. And the beauty of process-based models is we're entering this non-analog climate future, as someone mentioned earlier. So we can create, sort of these statistical models based on sort of what we see on the landscape today and how it matches to climate. But climate is changing in ways that we don't actually have a map to make today. So the benefit of process-based models is that you sort of understand how plants work and can use that sort of to better understand sort of how they might change with climate. So once I created that model, I'm going to actually validate it out in the field using this long-term ecological research record, specifically phenocams. So this is the range of red oak, and these are phenocams, which take pictures of the canopy every 30 minutes. So you can extract phenological information over very long timescales. I've gone out and I've actually sequenced all the red oak individuals growing in these phenocams using low coverage whole genome sequencing. And so I'm now working on putting all that data together, and I would love to chat with all of you about it. So yeah, if you have any questions, happy to chat now or anytime during this week.
How many,
Yeah.
In each of the sites, I couldn't see how many sites.
Yes.
It looked like 10. How many individuals are doing both of those sites, and how did you pick this individual?
So I've gone out and you can actually identify individual red oaks growing in the phenocam viewshed. So unfortunately, I only have about probably 12 to 15 per site. So I'm trying to figure out ways of using maybe the pheno network to sort of up that number. But the goal right now is to create this model, make predictions, and then actually check if that matches sort of what it looks like across the range of red oak. Yeah, that's sort of where I'm at right now with that part.
In some controversy, show that the maternal environment time of seed formation affects the phenology of the offspring. So do you think that happens in
People think so. I tried to work at the Arnold Arboretum basically because it's like a common garden. So I'm hoping that because they're in a common environment, that might help, but it's definitely a factor that may contribute.
Thank you.
Yeah.
Oh. So our next speaker this evening is Ami, and Ami is going to talk to us about transcriptomics of seasonal leaf senescence in a deciduous conifer, Larix larcina. I should have just said a larch species instead of butchering it.
Sorry. It's not here. It's not here. I can see it.
Vidya, can you help us?
It's not in the list.
It was here, it's in the downloads.
All right, thank you, everyone. My name is Ami, and I'm going to talk to you this evening about a study that I've been doing for the past couple of months called the transcriptomics of seasonal leaf senescence in a deciduous conifer. Sorry if my speech is a bit slurred. We just had a reception a few hours ago. So if you're like me, you might have already wondered how certain plants are able to like undergo certain changes, certain time of the year, giving us this very beautiful scenery of color changes in the leaf, and which usually ends up with the leaf being like upsized loss of leaves. And it doesn't happen in my home country, so I'm blessed to be this way. So this is what we call the seasonal leaf senescence, which is physiologically an aging process that is responsive to both environmental and internal cues. And so I am interested in finding the molecular changes that accompany this process, particularly the gene expression changes that are associated with the process of leaf senescence. Much of what we already know about this process is based on studies that have been done on angiosperms, particularly the Arabidopsis plant. But in this study, I'm interested to know if similar processes do occur in gymnosperm, particularly the Larix laricina, which is the species that I'm working with. And so we first collected the samples in 2 different locations within the state of Connecticut. And those collections were done at 3 different time points, which reflect the different stages of the process of senescence. So the first time point was when the leaves, or the needles, I should say, were green. And the second time point is when those leaves were yellow in color. And our final time point was just after abscission. For this study, unlike majority of other studies, we targeted the abscission zone tissue, not just because we wanted to do a different tissue, but also because if you look at the time point that I just showed in the previous slide, the 3rd time point reflects the process just after abscission. So we needed to look at the processes all through to that time point. So we targeted the abscission zone as the tissue for this study. And so we did extract our RNA and carried out sequencing using the Illumina technology. And after qualifying and quantifying our reads, what we first see when I plotted a PCA for the samples that I had, majorly if you look at the samples, all the samples on the left side, could be your right, I don't know. Yeah, that's your left. All of them are from one location, which we call location K. And all the other samples on this right are from another location called U. So location played was like a major factor in the distribution of the samples. But if you consider, for example, one single location, location U, you also see that the samples grouped according to the different time points or conditions. And all the samples in red are samples at condition 1, and all the samples in blue are condition 2, and green is condition 3. After carrying out a differential gene expression using the ESEC package, I was interested to see how the differentially expressed genes compared between the 2 time points. And so I compared time point 2 to time point 3, and I wanted to see how those differentially gene expressed those differentially expressed genes compared between the two time points. And what I see is that there is a similar pattern across the two time points, but largely what is clear is that time point 2 had a higher fold change for the same kind of genes, although we also see certain genes that have completely contrasting patterns of expression. And so again, just to recap on what I was talking about, this project uses a time course differential gene expression analysis with RNA-seq to answer fundamental biology question of the process of leaf senescence, how the gene expression changes across the whole process of leaf senescence. But it's also an evolutionary question where we're trying to compare what happens in gymnosperms, particularly the Larixalis in the species that I'm working with what we already know in the angiosperms. Here I have a photo of the Arabidopsis plant. So this study is being carried out at the Plant Computational Genomics Lab. That's at UConn, led by Jill. And I want to thank all the collaborators and the funders for this project. Thank you.
There's time for a wide question if it's a small one, a quick one. Okay.
Thank you.
It's loading slowly.
Yeah, as long as it's just full screen.
Okay.
Computer apparently had a little break too and had too much wine at dinner.
All right. Locked out of the room, locked out of the bathrooms, locked out of Kathy's talk. So our next speaker is Kathy Collingham, and Kathy's going to talk to us about differential integration of putative adaptive loci across the mosaic lodgepole jack pine hybrid zone.
Where's the full screen? Just the green one. Technology. It's almost there. All right, well, hybrids are fascinating and hybrid zones are fascinating. Hybrid zones allow integration of loci from one species into another. And sometimes those loci might be adaptive in the parental species and also become adaptive in the recipient species. And sometimes they might be neutral in the parent species and become adaptive in the recipient species. And the hybrid zone I'm really interested in is the lodgepole jack pine hybrid zone that goes through Alberta, British Columbia, and the Northwest Territories in Canada. And I've been working with a graduate student, Dania Remchak, on the question of how climate interacts with differential integration in this hybrid zone. And so to answer this question, we've got a slightly complex workflow that I've tried to break down into beautiful little pictographs. And so we started with a pretty large data set. So we have over 1,300 individuals genotyped at over 10,000 SNP loci. And those are SNP loci that were from the AdaptTree SNP chip. And we broke those into separate groups based on their species ID. So we ran them through structure to get at who was a hybrid, who was pure lodgepole, and who was jack pine. And then we took the lodgepole pine that was pure, and we looked for population structure, and we looked for SNPs that were associated with climate that suggest that they might be adaptive to those climate conditions. We did the same thing for jack pine, and then we subset our data, and we subset it into different hybrid zones and the parental classes only for the loci that had climate associated SNPs. And then we examined these climate associated SNPs using introgress and looking at the differential patterns of integration between lodgepole and jack pine in these hybrid zones. And then following that, we looked at the allele frequencies of these interesting loci across the entire landscape. So we used the full data set to make pretty pictures, because pretty pictures really do tell a story. And so for our species ID from our over 1,300 samples, we had 540 lodgepole pine, 669 jack pine, and 169 hybrids. And these are mapped on the landscape that you see here. And then from that, we didn't find any population structure in lodgepole or jack pine. We weren't surprised by this. But when we did the climate analysis and looked at SNPs associated with climate, we found 156 SNPs in lodgepole pine, and they were associated with things like elevation, mean annual temperature, temperature difference, and our H, which is relative humidity. And then for jack pine, we found some similar environmental associations, but in addition, we found number of frost-free days is really important, as well as climate moisture index and extreme temperature. So from that, we took these markers, so the 156 SNPs in lodgepole pine that were associated with climate and the 189 SNPs in jack pine, and looked at whether or not they were differentially integressed. And so we separated the data set into a northern set and a southern set of hybrid zones. And I'm going to call it the northern extent, and those are in pink symbols, and the southern extent in blue. And you might say, well, there's trees beyond those pink symbols, but if you're actually in the Northwest Territories, good luck finding a tree in those locations. So if you found a tree, you're lucky, but that really is as far north as the tree distribution goes. And so we examined patterns of integration between the blue and the red that were different. So we looked at the northern, we looked at the southern, and compared their patterns. And so this is even more complex, but I can break it down for you. So on the x-axis is the hybrid index. So 0 means they're jack pine, 1 means they're lodgepole, 0.5 means they're hybrid. On the y-axis on your left, that's the probability of the genotype, with 1 being the probability of the jack pine homozygote, 0 being the probability of the lodgepole pine homozygote, and 0.5 being a hybrid. And then we have the southern cline and the northern cline. And on the other y-axis, we have the number of observed individuals with those particular genotypes. And so I've pulled one of the markers that was significant in the north and the south. And when I say significant, there is a null expectation of what you would expect for introgression based on the parental allele frequencies. And this is in those dark green color blocks. And in the light green color block is the null expectation for a heterozygote genotype. And the solid line is our actual observations. And so when that deviates from that solid green, it tells us there's something different going on here, than it's not neutral. And the dotted line is our neutral expectations for our hybrid genotypes. So for this example here, we noted that in the south, we found more jack pine homozygotes and more heterozygotes with the jack pine homozygote than we would expect based on those parental allele frequencies. And in the south, we find less than. So it tells us that, you know, maybe in the north, where we have less, maybe it's not as adaptive in the north, and in the south, it's more adaptive. And so it's being selected for. And so we've found 28 markers of those climate-associated SNPs that showed differential patterns between the north and the south hybrid zones. And I've pulled a couple of examples to give you an idea of what sort of patterns we're seeing and what they might mean. So this pattern we observed in a number of loci, and this locus was identified in lodgepole pine, and it was associated with temperature difference or continentality. And what we found here with the clines was that the jack pine associated allele was introgressed in the northern hybrid zone. So it was introgressed into lodgepole pine. So we found a lot of jack pine type homozygotes in lodgepole pine. And you can see the red and yellow is really present in that northern hybrid zone and not in the southern hybrid zone. And we looked at the annotation thanks to Rhiannon's bioinformatic wizardry. And this locus is potentially associated with RNA processing and stress response. Another pattern that we saw quite often, and this was identified in jack pine, and it was associated with the number of frost-free days. And in this example, the lodgepole allele, so that blue color, is introgressed in the northern jack pine more than you would expect based on neutral expectations. But in the south, the jack pine allele actually looked like it was selected for in jack pine. So in the south, that allele is really important in jack pine, but in the north, the lodgepole pine allele is more important. And this locus is potentially associated with protein homeostasis, which could do all sorts of cool things. We can make up fun stories when it comes to annotation. And then the third example, this one was pretty fun. It was a locus again identified in jack pine, but associated with climate moisture index. And in this one, heterozygotes observed more frequently in the hybrid zone in both the north and the south. But it was really interesting to me because we see that the blue color is in both parentals, and the blue color means pretty much 0. And so this suggests that there's some sort of heterozygote advantage in that hybrid zone for this locus. And again, it's associated with signal transduction and perception. I mean, come on, let's drool over these annotations because they're so much fun. And so from this work, we're still kind of unpacking it and interpreting all of these clinal zones. But what we found is that hybrid zones are still really fascinating, and that we found some contrasting environmental differences between lodgepole and jack pines. So there's different environmental variables that are important in their adaptation, and that there is evidence of differential integration in this hybrid zone, and some of it is potentially associated with climate. And these sorts of information can be really important for understanding climate change impacts, forestry, reseeding, stock management, all of those really great things that we're interested in. And with that, I want to say thank you.
We have time for a question for Kathy.
Yeah, what's the range of genomic proportions between lodgepole and jack pine in the hybrid zone among individual trees?
It varies.
How does it vary?
It's a lot of it's, I would say it varies with the environment. And so in some regions, you'll find more lodgepole-like hybrids, and in some regions, you'll find more jack pine-like hybrids. There's a really strong environmental effect on what kinds of hybrids you're going to find.
If you find one that's more jack pine-like, its neighbors probably going to be similar.
Yeah.
And then that would be what, like 50-50, 25-75, what's sort of the proportional ranges?
It really varies. There are some really strongly back-crossed individuals, and we have detected F2s as well. So we've kind of got a good range of hybridity among our individuals.
Do you get F1s every generation?
We get F1s really rarely.
Oh, rarely? Yeah.
Rarely, yeah. In our samples so far. We're searching for them because that's what I want, more of those hybrids in these analyses. Yeah. Thanks.
All right. Our next speaker this evening is Laura. And Laura is going to talk to us about transcriptomic responses to WPBR infection in whitebark pine seedlings. Where's the map? Oh.
I'm never going to be asked to do this again.
So I have this. I can put it on. It is not there. So I'm going to present some preliminary results that we are finding on the response of whitebark pine seedlings to the whitebark pine blisters, and how this is. And this is a collaborative work with between Northern Arizona University and University of Yukon and the U.S. Forestall Services. I don't want to stop to things that you already know. And what whitebark pine blisters is a very difficult and the worst, one of the worst forest disease across the northern hemisphere, and can affect the 5 different species for 5 leaves, the whitebark pine species. So our goal in this experiment is identify the early response after the fungal infections in seedlings and recognize the impact and resistance characterization of Granatium rubicola infection. In order to respond to this, we are working with 6 different half seedlings growing in veterinary researches, forest researches that came from a mother tree that has been shown signals of resistance in several previous infections. These 6 seedlings has 2 years growing, and we expect to recognize. With the design experiment, it is 3 different replicates that has been inoculated and 3 other individuals that has been not inoculated, working as a contract, and we extract the RNA response after being inoculated in this tree and process that to recognize the cDNA that has been and go to Illumina technology to recognize the transcripts and assembling this transcriptome to construct a global reference, and after that recognize the different expression analysis between the 6 individuals and to know the functional attention of these genes and the differences according to this comparison. For the global reference that we construct, we found 25,000 genes, different mutation, and we have like 61% of the boosted genes already found, and the assembly was constructed by Trinity and recognized the gene families annotations and the pipeline, the bioinformatic pipeline. For the gene families that we found from the 3 individuals that has been inoculated, we found an activation, a very active response of the COS response of biotic factors that involves the response to osmotic stress and the defense response to another stress that has been already published. And we detect an increment of the transcriptomic revelations of much of several metabolites, and this analysis was conducted by OrthoFinder,
And for the different expression analysis, we identified three differential genes that were 56 was positively regulated. That involves the response of immunity level of the seedlings that activate several abiotic factors and stress regulators as that involves transcriptive factors for and repressors of sorry, I am just questioning here. Transcriptors transcription factors that are involved in the response to stress as MIPS factors and 27 genes that are downregulated that are involved in the reduction of productions of metabolites like organic compounds. The next steps for these samples will be, we are trying to identify the genetic and the metabolic pathways that are involved in these genes and characterize the genes and the gene families that are involved in the resistance if these individuals has been selected as potential resistance organisms, and construct a set of annotation of the white barn genome, and also explore how different could be the methylome of these 6 individuals. And with that, I will add to end to thank all the funding and all the collaborators that are in this project.
Thank you. I'm sorry if I missed this, but the 3 replicates that you had for the inauguration, are they clones? Are they the same family? What were they?
There are 6 members of the same family, so they are half siblings. It is, yeah, they become from the same mother tree, and yeah, as there has like open pollination, so we don't know where.
Thank you.
Our next speaker this evening is Kriti. And Kriti is going to talk to us about using transcriptomics to annotate and classify potential disease-resistant genes in white pines. Oops.
Hello, everyone. I'm here. My name is Kriti. I'm here to talk to you about my work on identifying and classifying disease-resistant genes in white pines. And so I assume that most people here are probably familiar with pines, and I guess the white pines are the subsection Strobus of the genus Pinus, and they're species native to North America, Central America, Asia, and Europe. And so there are 6 species native to the US and some species native to Mexico as well, and Canada. And so these are all incredibly ecologically important, especially the high elevation pines that often mark like the tree lines of Western mountain ranges. And so why do we care about the white pines? Well, I know some people have talked about this before, but briefly, because of white pine blister rust, it can infect all species of North American white pines. And it is especially damaging to seedlings. While it may cause mortality in adults over time, it causes high mortality in seedlings, and it is spread. It is caused by the fungal pathogen Penitum rubicola, and is since spread throughout most of the native range. And so, in combination with like changing climates and pests such as the mountain pine beetle, this is leading to devastation of a lot of Western pine forests. And so plants aren't completely defenseless against disease. They have disease resistance genes of the type known as nucleotide binding leucine rich repeat receptors, or NLRs. And these consist of a characteristic structure with an N-terminal domain, a nucleotide binding domain, and a leucine rich repeat. And these domains can actually be used to identify potential NLRs within the gene space. And so in order to do that, I first need a representation of the gene space. And so from a recent phylogenomic analysis of pines, I was able to obtain RNA sequencing data for 22 species of white pines. And here the graph shows the original geographic distribution of the species. And I have species across Asia, North America, North and Central America, and Europe. And so here's just a phylogenetic representation. And really what I want to focus on here is the circles in red represent susceptible species, and circles in blue represent like more species that are known to have resistance, specifically the Eurasian and Asian white pines. And so the process after assembling the transcriptome using these RNA resources, I used both the protein sequences and the coding sequences, and 3 different methods to identify these NLR type genes. InterProScan and RJ-Augury directly scan the protein domains, whereas I also use NLR Annotator to look at the motifs. And so after combining and filtering this to create a more representative set, I have now my set of predicted NLRs. After predicting them, I then got to classify them. And so there are 3 major subfamilies within the NLRs, the TNL, CNL, and RNL, and these are differentiated by their N-terminal domain, but also certain motifs within the NDOC domain. And then I also classified or identified what percentage of transcripts were complete. And to be considered complete, they had to have all 3 domains. And so here is my results. I was able to identify 6,784 potential NLRs across the 22 species. The bar graph on the left shows the distribution by type or classification. And as you can see, there are a lot of NBS and TX types, which were 2 of the largest incomplete categories. And so looking over at the pie charts on the right, you can see that only 19% of the transcripts identified could be considered complete, or the NLR transcripts I identified were complete. And only 56% of them could be classified according to their N-terminal domain. And of the ones that could be classified, here on the left side, I have the distribution across the classifications. In my results, the TNL category was largest. However, unexpectedly, I was not able to identify as many CNLs as based on a prior study across multiple conifer species. And then here, looking across the phylogenetic representation, you can see a little bit that the Asian species were able to identify more. I was able to identify more CNLs within those rather than compared to some of the North American and European species. So it'll be really interesting to look more closely at genes that might be shared or contrasted across these species. And so as an overview, I was able to combine different NLR identification methods. There are multiple software that exist and a methodology that I created as well. And so this allowed for a more complete representation of the potential resistance gene space, especially because certain domains could only be identified by certain programs. I identified thousands of potential NLR genes that can then serve as a resource for annotation. And then some of the challenges and caveats with working with the transcriptomes were differentiating like presence absence, doing comparisons across species has been a little bit difficult. And so some of the future directions is really trying to look into orthogroups and see what genes are shared in those orthogroups and if those are any connections that can be made. And I would like to thank everyone for helping me. And don't have it on the slide, but also Dr. Richard Sinisco for all of his help as well. And Dr. Rigson and the plant competition biology lab. Okay.
Thank you.
Question for Jackie? Oh, sure. Thank you.
Just within a species, how many resistant genes? I guess you're defining them basically with the NOR. But how many within a species? Because across species.
There was a range. So, and it ranged from around like 200 per species to I think around 700 per species. So there were some species that had more NLRs identified, but overall, like the proportion of complete and the portion that could be classified was roughly consistent.
And with your methodology, did you tell which ones were in duplicated blocks? How many camera repeats would you have? And how many were?
Unfortunately, with the transcriptomic resources, I can't. I can identify like, I guess, transcripts that are identical, but like not really where they would be located in the genome. But hopefully with more genomic resources, we will be able to do that.
I'm going to break all the moderator rules and ask an additional question. So do you think that the ones that you found that were incomplete are because they've dropped whatever motif is there, and so they're no longer doing the same function, or because the transcriptomes aren't that complete? Like, which is it both that's causing some of that?
I think it might be both. I definitely think that the incompleteness of the transcriptomic resources is a really big challenge and caveat with a lot of my results. But based on other like research that I have found, there has been some people do claim that these truncated NLRs may still be functional, which is why I included them, because we're not as certain as to whether these truncated copies are functional, but they are being expressed if I'm finding them in the transcriptome, which is also why I kept them.
Yeah. Okay. Thank you.
And our final speaker closing out this awesome day of science is Haley. And Haley's going to talk to us about genetic diversity in BC reforestation, a closer look at effective population size.
Where are my slides? Oh, they're over here. Yeah.
And then you have to like.
Okay. Well, thanks everyone for sticking out until the end. I already really like this conference, but everyone being here makes this my favorite conference ever. I want to start off by acknowledging that UBC sits on the traditional and unceded territory of the Musqueam people in what we now call Canada. And I'm going to talk to you, like has already been said, about effective population size in BC reforestation. So here in BC, the generations are starting to increase in the selective breeding programs at the same time that the percentage of landscape that is harvested and replanted with the selective seed is increasing. And it's important to remember that these landscapes are a mosaic of planted and natural stands. And so this has led both managers and the public alike to ask what the consequences of this forest management is for landscape level genetic diversity, especially because diversity at this scale will be incredibly important for resiliency to climate change for these forests. And so the first step in addressing this question is how well is genetic diversity maintained within the breeding program? And here in BC, it is monitored and maintained at the seedlot level by estimating effective population size from parent tree contributions in the seed orchards. And so we want to know, how do these estimates compare to genomic estimates? So how are these boots on the ground estimates comparing to genomic estimates? As well as how genetic diversity in these selectively bred seedlots compares to genetic diversity in natural seed. And so we address these questions by looking at 2 BC forestry species, lateral pine and interior spruce. And I was lucky because there's already some great genomic data sitting there, which is your dream as a postdoc. And so we looked across panels of roughly 1,200 putatively neutral and genome-wide SNPs, comparing both selectively bred lots as well as natural lots, so seed that's taken out of these natural stands. And we compared genetic diversity estimates in natural stands versus selectively bred lots.
as well as comparing different estimates of effective population size. So we have that boots on the ground estimate using gamete production, and we compared that to 2 estimates from genomic data, one using linkage disequilibrium and one using SIB chip frequency. So looking at the number or frequency of siblings from pedigree reconstruction. And the results were quite similar across interior spruce and lodgepole pine. So I'll just be showing you the results from lodgepole pine. And so when we look at these typical estimates of genetic diversity, expected and observed heterozygosity and inbreeding coefficients, and we're looking across natural in green and selectively bred in blue, we see that the estimates are quite similar and that they are not significantly different. But these differences start to really emerge when we look at effective population size. And so on the left, you will see our selectively bred lots. And I'm not showing you the natural lots because most of the estimates were either at infinity or approaching infinity. So this is where we really start to see differences. And then when we look at that second bit where we're comparing the different estimates of effective population size, we see that our gamete production estimates in orange are quite similar to our 2 genomic estimates in green and blue. And most times they're actually underestimated, which is really great news for genetic diversity. It means that what's going on in the landscape is more than what had been the minimum for monitoring. And when we look where that gamete production estimate is less similar and we look into the characteristics of our seed orchards, we can start to understand why. If it's underestimating, it could be down to pollen contamination based on where that seed orchard is located. And if it's overestimating, it could be down to forward selection and the equations that are used to calculate that effective population size. So now we have a pretty good understanding of how genetic diversity is maintained within these breeding programs, but that does not fully address the question of what the consequences of forest management are for genetic diversity at this broader scale. And so the next step is to look at the shift in genetic diversity from orchards to seed lots to actual planted stands on the landscape. And then we'll be using the results from step 1 and 2 in a landscape genetic simulation model, modeling these natural and planted stands that are interconnected through gene flow on the landscape and varying parameters that are associated with this forest management in order to look into the future and really understand the effect of forest management on genetic diversity at this broad landscape scale. So keeping it short and sweet. If you have any questions, let's chat. Come find me or email me or tweet at me. But if you have any questions, comments, or just really like conservation genetics, I'm down to talk. And I would really like to acknowledge all of my great collaborators. Sally Aiken is here. And this is an incredibly collaborative project that is tightly linked with both industry and the Ministry of Forests in BC.
Do you have any questions for Kathleen? No, I'm failing as moderator, not thinking of it.
That's fine. It's quite late.
We spoke about it a lot.
Yeah, we did.
Could everyone join? Oh, do you? Oh, sorry.
So, what's your recommendations to the tree breeders in those 2 species given the results that you see so far?
Yeah, so that's a really good question. I was expecting someone to ask, and we're still in that discussion phase because it's not obviously Sally and I giving recommendations to the tree breeders. It's us working with the ministry and getting input from the breeders in order to come up with what the next step is. And so, yes, we now know that unsurprisingly, the effective population size is lower in these selectively bred seed lots. But what does that really mean? And so I think that the simulation modeling is going to really help us better understand what the true consequences for that are. I mean, BC already has a minimum NE, which is great, ahead of the game in a lot of ways. And so whether or not that minimum NE is like it's a good minimum is really going to come at the end of this project.
Okay, could everyone thank Haley and the rest of our speakers this evening for giving us these great talks?
Thank you, everyone, for a great first day. You're going to get a completely new program for tomorrow because we have a few changes. Not that new, but everybody will be in the general same order, but we're going to shift things around a little bit. So look for that in your email tonight. But the most important thing is we start at 8:30 tomorrow morning and not at 9. So please keep that in mind, and I'll send something out for that after.
Thank you all.
Yeah.